A very good morning to all the participants. We will start the program in a few minutes from now. Dear participants, I kindly request you all to mute your audio and video for better bandwidth and reception. Thank you. Good morning. A warm welcome to all the participants on this pleasant morning. On behalf of the principal and the organizing committee members of the PG and Research Department of Zoology, Dr.Ambedkar Government Arts College, Chennai, we indeed delighted to begin the 3-day National Capacity Building training on marine biodiversity and its conservation. This program has been designed to focus the major aspects of marine biodiversity and its conservation, and it is divided into 3 different teams for all the 3 days. So I think this 3-day program will help all the faculties and the research scholars to have a training on the marine biodiversity and its conservation measures. We have 4 eminent speakers today. Dr. D. Adhavan, Scientist C, Center for Climatic Studies, Satyabama Institute of Science and Technology. Dr. Titus Emmanuel, Research Assistant, Marine Biological Research Center, Zoological Survey of India, Chennai. Dr. M. Arumugam, Associate Professor, Faculty of Marine Sciences, Annamalai University, Chidambaram, Parangipettai. And our last speaker of the day is Dr. K. Mahesh Kumar, Assistant Professor of Aquaculture, PG and Research Department of Zoology, Government Arts College for Men, Nandanam. So these are the 4 speakers who are going to present their presentations today. Now, I request our head of the department, Dr. T. Kumaran, to deliver the welcome address. Over to Dr. Kumaran.
Very good morning to one and all. With the blessing of Almighty, I welcome one and all. I am Dr. Kumaran, Head PG Department of Zoology, Dr. Ambedkar Government Arts College, Chennai. It is my great pleasure to welcome you to the National Capacity Building Training on Marine Biodiversity and its Conservation. I first wish to extend our warm welcome and greetings to this 3 days program. We have privilege of holding our 3 days program under the leadership of our principal, my dear colleagues, and especially organizing secretaries. Biodiversity is 2 words, biological and diversity. It refers to all the variety of life that can be found on earth, that is plants, animals, fungi and microorganisms, as well as to the community that they form and the habitats on which they live. Biodiversity is important to humans. It provides functioning of ecosystem. There are 3 types of biodiversity, namely genetic diversity, species diversity, ecological diversity. Marine biodiversity therefore refers to the species richness and abundance in the world's ocean and seas. And since the world is covered with approximately 70% of water, the amount of life in the ocean is enormous. Healthy marine ecosystem are important for society since they provide service including food, security, feed for livestock, raw materials for coral rock and sand, and natural defenses against hazards such as coastal erosion and deliberate flooding. At least 40% of the world economy and 80% of the needs of the poor are derived from biological resources. In addition, the richness of the biodiversity in life, the greater the opportunity for medical discoveries, economic development, and adaptive responses to such a new challenges as climate change. Conservation means preservation, protection, restoration of the natural environment. We know that human beings are totally dependent on nature. We can save the biodiversity in 10 things in our mind. Mind your carbon footprint and reduce the energy consumption. Make safe, sustainable seafood choices. Use fewer plastic products. Help take care of beaches. Be an ocean-friendly pet owner. Support organizations working to protect the ocean. Don't purchase items that exploit marine life. Influence change in your community. Travel the ocean responsibly. Educate yourself about oceans and marine life. Reduce, reuse, recycle, or waste to save the nature. No man is island. In any given occasion, there will be always be people who accepted so much effort for its success and even cannot be successful without people like our principal, staff members, organizing secretaries, and not but the least participants. I thank one and all. Stay home, stay safe. Jai Hind. Vanakkam. Over to Saravanan.
Thank you, Dr. Kumaran sir, for emphasizing on the importance of how we have to save marine biodiversity and what are the measures which are needed for the conservation of marine organisms. Now, it is over to Dr. N. Tirunavukarasu, Assistant Professor of Zoology, who will be focusing on the theme and the need for this research capacity building training on marine biodiversity and its conservation.
Good morning, one and all present here. Now I'm going to give a hint on why the theme has been selected and what is the focus of the today's program for the National Capacity Building Training on Marine Biodiversity and its Conservation. India has 8,129 kilometer coastline, 2.02 million square kilometer of exclusive economic zone, having nearly 2,000 species are recorded in Indian seas. It provides food, medicine, materials, fishing, and tourism. We have wonderful and different ecosystems such as estuaries, lagoons, backwaters, salt marshes, rocky coast, sandy beaches, coral reefs, mangroves, and islands. We have various hotspots, includes Gulf of Mannar, Park Bay, Gulf of Kutch, Gulf of Combe, Andaman Nicobar Islands, and Lakshadweep Islands. Today, the marine ecosystems are deteriorating at an alarming rate. The factors are overexploitation of species, introduction of exotic species, pollution from urban and agriculture areas, habitat loss, and decline of water quality. Keeping in mind, the National Capacity Building Training focused on disseminating knowledge on conservation of marine biodiversity to the community of students, faculty members, scholars, and stakeholders. The eminent subject experts are going to present their respective fields for the 3 days program. The major theme includes diversity and conservation of invertebrates, mainly focusing on seagrass ecosystem, corals and sponges, marine mollusks, and echinoderms for the first day program. And tomorrow, the second day, the conservation of diversity of invertebrates includes ornamental fishes, sea turtle, and dugong marine mammal. The last day, 31st of July, we have the program on the main theme includes climate change and ocean acidification, parkway conservation, and endangered marine animals. So the resource person in the program are mainly the youngsters at the average age of 37 who are working for nearly 10 to 15 years. Dear participants, have a wonderful training experience and conserve our mother nature. Jai Hind. Thank you.
Thank you, Dr. Tirunavukarasu, for giving the focal theme of this program on marine biodiversity and its conservation training. Now we move on to the technical sessions. So our first session today is going to be presented by Dr. D. Adhavan, Scientist C, Center for Climatic Changes Studies, Satyabama Institute of Science and Technology, Chennai. Sir is going to present on the sea grass ecosystem. Now I request Professor Malai Chelvi, Department of Zoology, Dr. Ambedkar Government Arts College, to introduce our resource person, Dr. D. Adhavan.
Welcome to one and all. It is my immense pleasure to introduce our resource person, Dr. D. Adhavan. Scientist C, Center for Climate Change Studies, Satyabama Institute of Science and Technology, Chennai. Mr. Adhavan completed his PhD in Ocean Studies and Marine Biology at Pondicherry University. He was a recipient of grants and awards from the Rafford Small Grant for Developing Technology for Successful Transplantation of Seagrass in India with the respect of climate change. And he was also received a Best Research Paper Award, Madras Christian College, Madras. Globally, he was working as Program Coordinator, Marine Biodiversity Program at Tridel Maritime Services, LLC, UAE. A couple of months, he was working as consultant Indo-German Biodiversity Program. Then, project scientist at Wildlife Institute of India, Dehradun. He was also served as marine biologist at Marine National Park, Gujarat Forest Department, Gujarat. Apart from this, he knows scuba diving experience in Andaman, Gujarat, Abu Dhabi, almost all Indian coastal areas. He was wrote 2 books and 4 training manuals. For these 4 manuals, 3 manuals related to coastal marine biodiversity and 1 for IFS officer training manuals, nationally and internationally. He was published 17 research papers in various reputed journals. His area of interest includes benthic survey and marine habitat restoration at Gulf of Mannar and Palpe, bycatch assessment, intertidal biodiversity assessment, conducting training and workshops on coastal and marine biodiversity, capacity building to forest department officials and local community. Now, I invite our eminent and excellent resource person, Dr. D. Adhavan, take over this session to share his ideas. Thank you so much.
Thank you for introducing me, ma'am. Share this.
Dear participants, a small request. I request you to mute your audio and video for better bandwidth and reception. Thank you. Dr. Adhavan, now
the screen is shared, sir.
visible, sir.
The screen is visible now.
You can make it as a slideshow.
Is
Yes,
it okay, Sana?
yes, you can start.
Yeah, okay. Good morning, everyone. And I'm very much thankful for the management and Department of Zoology, Ambedkar College for this opportunity. And I thank everyone for this introduction. And today's the topic was like seagrass ecosystem and its conservation importance. First of all, we see the coastal ecosystems. The coastal ecosystems are all are interlinked, and every ecosystem are important. This ecosystem includes salt marsh, mangrove, wetland, estuary, and seagrass, and all. And among them, there are three key ecosystems, key habitats, that is mangrove, seagrass, and coral. Why we are calling this key habitat? Like they play very significant role in climate change impact and revenue generation and providing ecosystem for other organisms. And today we'll be seeing a seagrass ecosystem. First, what are seagrass? Seagrasses are marine plants. They are only flowering marine plants, and they have long, thin leaves. And they have a different shapes of leaves, so like ribbon or paddle shape or oval shape. That is depends on species. And there are approximately 50 species worldwide. And science believes that the seagrass evolved from terrestrial before 75 to 100 million years ago. And we can see the seagrass in the shallow area and in the estuary, salt swamps, and intertidal region as well. And when we visit the coastal ecosystem, many people they used to mistake seagrass and seaweed. So first we'll see the difference. What are the difference between seagrass and seaweed? Seaweed, otherwise called as marine algae, macroalgae, and these seagrass they have leaf, flower, rhizome, root, but seaweed or marine algae they doesn't have root or leaves or flower. It is totally different. And a species from Colopar resembles like seagrass. Like many people they misunderstand the seaweed with seagrass. And the sea algae, marine algae, they have a blade and hold fast. Right? They what they do is they'll hold any hard substrate, rock or something for their root. But seagrass will have a root like proper root, so like proper grasses in the terrestrial. Uh, and the importance of seagrass. The importance of seagrass is they provide a habitat for various organisms, right from small microorganisms to megafauna, including dugong, sea turtle. And seagrass is plays a significant role in maintaining water quality. It is also called the lungs of the sea. And coastal protection. Of course, the wave action will be reduced, and the wave or current action will be maintained by the seagrass ecosystem. And the fishery production. It is very important for fishery production because most of the fish, they provide seagrass ecosystem provide a nursery or breeding ground for many commercially important fishes. So fishes they'll lay the egg in seagrass ecosystem so that they can avoid predators and the spawning ground for many organisms, right from crustaceans, crabs to the fishes. And economic importance. If you see the seagrass have very good source for tourism, how it is playing a role in tourism. Like for tourism to grade the beach, two things are important: the color of the sand and the water should not have any coliforms. So what happens is when the river runoff comes from the land, the this coliform or any polluted water, they are filtered by this set of ecosystems, salt marsh, mangrove, seagrass ecosystem, and then the water goes into the sea. So this is all this ecosystem, including seagrass, they play a very important role in maintaining the sediment and the coliform and controlling the coliform bacteria. The all the beaches they should have clear water. Only then it is fit for tourism, then it is great. And livelihood for coastal community, for fishes, and it is also a storehouse of biomass of a lot of organisms. And we can next slide we can see the picture of how the seagrass is playing important role. These seagrass, they are like good source for carbon sequestration. They see as they sequester carbon and they filter sediments and other nutrients so that they can keep in check of the algal bloom. And they also provide ecosystem for fishes and reduces the sediment resuspension. How they reduce sediment suspension is like their you can see next slide that one. Yeah, this seagrass they are binded to bottom layer and the leaf of the seagrass they have a baffling effect along with the wave. What happens is the moment of the blade of the seagrass, they hit upon the floating sediment, the loose sediment which is coming from coming along with the water which is carried through other ecosystems, salt marsh, mangroves, and like wetlands. These floating sediments are hit by the leaves of the seagrass, and they got they fall down and settle down. So the set of organisms they they play in the digestion of these sediments or nutrients or other impurities. And yeah, this is important role in maintaining the water quality so that the water the ecosystem will be clear and the next ecosystem like the clear water will be passed on to coral reef ecosystems. And yeah, so the research phase is effect plays an important role in sediment stabilization. And this is the feature representation. This seagrass, they in the first picture they provide ecosystem for many fishes. Like if we have a seagrass there, then we have a rich biodiversity as they provide a shelter for many organisms and hideout for many small juvenile fishes and the commercially important fishes. And in the half of the thing, like we can see that there is no seagrass. If there is no seagrass, the diversity will be reduced. And the next place, what we see is the baffling effect of the seagrass blade. When the floating sediments they are released into the marine environment, this the baffling effect of the waves and along with the seagrass blade, they hit down the sediment and they settle down. So the set of organism in the root or epiphyte, they make use of those sediment and nutrients and they digest that. And the next one is the main role, main effect of this mangrove and seagrasses. The organic carbons, particular organic carbons and inorganic organic carbon during outfilling, they are filtered by the mangroves and the seagrass ecosystems. And they also play an important role in damping the wave, reduce the wave action. As we said that before, they are also playing important role in trapping the sediments, so that sediments and nutrients, so that the algal bloom will be in control. And trophic cascade, they are playing important role in transferring the energy from one trophic level to another trophic level. And this is how the seagrass works. Seagrass set of cell as chloroplasts, they utilize sunlight for photosynthesis process, and they also use minerals which is from the adjacent ecosystem, and the nutrients settle down in the bottom and it is absorbed by the roots of the plant, and they release oxygen. They one square meter of seagrass ecosystem produce 8 liters of oxygen per day, and they also they are very much very important source for the carbon sequestration, trapping the blue carbon and storing into the sea, into the sediment. And nearly this seagrass, They are sequestering 24 TG of carbon per year per hectare. And the major seagrass ecosystems in India. We can see the seagrass ecosystems in Gulf of Kutch, Lakshadweep, Park Bay, Gulf of Mannar, Chilka, and in Odisha and Andaman Nicobar. Apart from this place, seagrass are also sparsely distributed in other coastal places like Maharashtra, Goa, Karnataka, Kerala, and Andhra Pradesh. It is sparing distribution, but the major seagrass ecosystems are these. Gulf of Kutch, Lakshadweep, Mannar, Park Bay, and Odisha, Andaman Nicobar. And these are the seagrass species what we have. We have nearly 16 species of seagrass and approximate cover of 500 square kilometer. And on the table, we can see the number of species which is found in each and every state. And threats to the seagrass. There are various threats to the seagrass ecosystem that include natural as well as man-made. Natural means like due to the climate change and huge hurricanes and all. And plastic pollution again. First picture like this is like plastic polluted by the fishermen and the boat making place. And these plastics and other things are thrown into the sea. And even the domestic waste is also released to sea, and it causes high nutrients which they spread to the seagrass ecosystem. And fishing practices, dragging and prawn net. These practices in the seagrass ecosystem are really an impact for the seagrass ecosystem, negative impact for the seagrass ecosystem. And we can see a lady is collecting shell in the seagrass ecosystem by digging it. This also that is for her livelihood. And anchoring of both the seagrass and coral ecosystem is a really we can see this in Mandapam, Legacy Modern Farm area. They are anchoring the boat and using fishing gear in seagrass and coral ecosystems. And apart from that, fishing practice, like as we said before, trawl net is one of the major. The major thing is fishermen are not aware of the importance of the ecosystem. And second is plastic pollution. The fishes plastic pollution is also another threat to the whole coastal ecosystem. And the releasing of sewage water. This high nutrient level or this toxic sewage will decompose the leaves of the seagrass ecosystems. Yeah. And adaptation. Adaptation of the seagrass is like the normal seagrass, they can withstand from 6 degrees to 40 degrees centigrade. And they have roots. They have horizontal stems called rhizomes, and it is like the roots are buried in the bottom, and so that they have some air vacuum in the leaf. So the leaf will be erected towards the surface into a stalk. And and these are flexible to withstand the current and the underwater current with wave action. And if you see the mixed seagrass colony, the short seagrass like ovalis and holidays, Immunovers, they are short and they grow fast and they stabilize the sediment for the taller seagrass, and which they grow slowly. And so these seagrass they stabilize the sediment and they take the nutrients, so that the other seagrass also will be utilizing these nutrients. Okay, how does seagrass grow? As as we know, like seagrass also need nutrients. These nutrients are taken from the other ecosystems by the river runoff, and and they also need sunlight for the photosynthesis. And the seagrass they cannot grow in the dry place during the low tide. If the place is dry, they cannot survive there. So they need water. And the seagrass they always grow in the shallow water near the shore, and the water should be calm. And seagrass makes the water clear, and it is to for its survival. It is also called natural engineer. And if the water is too much turbid due to like human impact or coastal development, then there are chances for the seagrass to get dumped, die. Because this over sediment or the sudden dumping of mud will really cause a negative impact on the ecosystems. And seagrass leaves grow fast and rhizomes they develop slowly. That is the reason like once you get rooted, it will take a long time to get stabilized and grow. And how they reproduce. Seagrass they reproduce by sexual as well as asexual method. First is like they detach from the mother, the colony, the shoot will be detached, and it is transferred by the wave action or current to the other place and and it will get stabilized. And second is the rhizome fragment. If the rhizome is fragmented, then it is washed out by the current or wave action, and it can be settled in some other place where the climatic and where the environmental condition is favorable for the seagrass growth. Then it can settle there. And again, the viviparous propagation, the whole fragment of the fragment of the other seagrass will be detached and will be going to other place and get settled. And based on this, principles methodology, the restoration techniques also followed. Uh, uh, and next is the sexual reproduction. The sexual reproduction is as we know, seagrass has the longest pollen grain in the world compared to other plants. And so the male seagrass leaves the pollen, and it is taken by the water current or any animal life, same like in the terrestrial. There are some carrier for the pollen grain. The recent research they said a crab associated with the seagrass ecosystem is carrying the pollen grain. So this seagrass, the pollen grain will be taken to the female, and when it meets the female seagrass, it will reproduce. And after reproducing, how it forms is the fruit or seed is released into the water. Then it will go to the suitable place through water current or wave action. Then it will get settled at the place where it is suitable. Again, the research says in the stage 3 we can see based on the different terrain like the bottom the seagrass they have different adaptation. In pebble they have a set of adaptation, and it's a sand. The second picture shows the sand adaptation and the fiber, how the root get adapted, get anchored, and they stabilize to grow. And the seagrass restoration process. Based on this, the effect the previous what we seen in the previous thing, how it propagate on vegetables. The restoration technology is also followed based on that. The restoration of seagrass is followed because due to the loss of seagrass in many places. That is due to coastal development and unregulated fishing practices and other processes. And transplantation, transplanting vegetative rhizome is commonly used practice worldwide. And collecting the pollen grain is another technique, but it is not commonly used in many places because this one will be feasible and for the transplantation process to the for restoring. And restoration is it is always the second major thing to, after conserving the present ecosystem. Why they are restoring is during the if the seagrass are destroyed recently due to unpredicted weather conditions, but still the environment is conducive for the seagrass ecosystem, then the restoration can be carried out at the places. And there are different methods followed by Fonseca et al. For the restoration. First is plug method. In this plug method, what they do is they'll collect the seagrass with the instrument which will be like a plug, and the whole seagrass that part will be taken from that place, and it is transported in a plastic tray, and they'll go to the place, the recipient side, and the recipient side also will be dug with the same shape of that the plug of cigarette what we taken from the donor side. And uh, uh, this that transplants will be placed in that places, so that the seagrass will be stabilized and it will be growing in that place. And like this method will be suitable for shallow water where the underwater current is less. And the staple method. Then the staple method is something like what they do is they'll take the rhizome and the seagrass, and from the donor side, the seagrass recipe, seagrass with the uh, rhizome is taken, and again with the help of the plastic tray will be transported to the recipient side, and the seagrass along with the rhizome will be tied up with one string, and a metal clip will be used to place this rhizome seagrass inside the mud. And again, this method is also like it is feasible where the current pattern is less, because if there are chances for this newly planted seedlings to get unplugged due to movement of water current. And next is feed pot method. Feed pot method is what we use in the garden, same like that. We use to take the feed pot, feed. In that feed, they used to make in like a shape or square or round shape. They then dried, and after that, the seed and the metal stem will be taken and plugged inside that. Then it is taken to the recipient site, and then it is planted. All this method are followed different places, and uh, based on this method, we also adapted a technology to restore seagrass. This project was funded by Ruford to restore the seagrass like at the South Touch area. And actually, this was experimental work carried out at this place. And first of all, the objective of my work was to study the distribution of seagrass and select the site for the nursery development and restore the seagrass ecosystem. And we selected these places, in Gulf of Kutch, we selected Metapur area. Why we selected Metapur area is as this is an experimental work, so we need a place where the flow of current is less. As we know, Gulf of Kutch is highly influenced by current, and but this place is very less influenced by the current because this is out of the Gulf area. If anything happens, any place inside the Gulf is on, it is influenced by the sediments as well as heavy current pattern. So this place was selected, and three places were selected based on their like their availability of seagrass and current pattern. First of all, we assess the seagrass ecosystem at this Gulf of Kutch area, and we see that three places we assessed: Narara, Kirotan, and Metapur area. From this, we have uh, recorded Halophila ovalis and Halophila uninavis were abundant when compared to other species, because at this Gulf of Kutch is a dynamic place where the sediments will be keep on moving. So the diversity of the seagrass also will be keep on changing from season to season. Okay, and we selected these species like Halophila uninavis and ovalis, Halophila ovalis. As this place was fully diversified by this seagrass ecosystem, as well as there was a recent disturbance of this ecosystem by the cyclone, and so this species also preferred by dugong and sea turtle. These are the major reasons we selected these species. And the site selection. Site selection, we selected the place near a reef structure where again this reef structure will hinder the current pattern, the water movement, so that it will have a hideout the current pattern will be reduced. The current force and water movement force will be reduced by this reef. And it was nearly a 4-meter depth where the like during the low tide, and the seagrass like the new recruitment of the seagrass is also like is a positive sign for our like experimental work for the light intensity. So we understood that there will be enough light for the seagrass also, and the like the particles of the seagrass are transported for this work. And before transplanting, we have checked the the water quality, temperature, pH, specific and salinity for 1 year. It was regularly monitored. And the methodology. Yeah, first method actually we use the staple method as we seen before. The vegetative part of the rhizome will be taken from the donor side and will be placed in the recipient side with the help of a paper clip or any metal clip to secure the rhizomes. So this what happened was during our work we transplanted this on one full day, and when we checked the next day, this all these transplants washed away due to water current and this thing, underwater movement. So what we did is we did slight modification with this staple method. What we did is we constructed 1 meter by 1 meter square mesh iron frame with a mesh inside that iron mesh. In that, the 4 corners were anchored or it is tied up with the boulders so that this frame will not come outside. And with this, the shaken meristem will be taken, and it is along with the mesh, they are kept inside that frame, and they are tied or clipped with the mesh along with this mesh, so that we made sure this this meristem or the new these seagrass are not washed away by the water movement, so that this metallic cage or metallic uh, uh, frame that produced supported the seagrass during the high water current movement. And the plantation was carried out in consecutive days. And these are the methods, these are the things we use. This is the frame, a metal frame, and metal mesh were used inside this. And this is before doing the transplantation, we placed that, and after placing this place is also acted as aggregating device for many fishes and other couple fish and all. It was visiting the place, and it we felt it is a positive sign for the diversity. As the main role of seagrass is providing habitat for many organisms. So this was a positive sign, and yeah, and this is the methodology. After placing this, we use it seagrass meristem and we placed our inside the metal mesh, and we tied up with the metal string. We use only metal, not the plastic, so that the electron when it is degraded, it will go off and the seagrass will be stabilized with the help of scuba diving. And we were monitoring this in consecutive weeks, few weeks, the same. We found the seagrass was stabilized well and it was not washed away during this underwater current or any water movement. So we found this methodology was adaptable, was good enough to support the seagrass vegetation transplantation in Gulf of Kutch area. So in other places also, based on the terrain or water current, we have to modify. We have to go for slight modification of this methodology. And unforeseen difficulties, what we see Vitapur again, Gujarat is known for turbidity. So during many times we have seen the visibility was poor, and the weather was also not conducive. It was very difficult for us to travel to the place. And iron frame, many of the iron frame were washed away due to underwater current, during the tropical cyclone that is like really a great loss for this project. And as we said before, like for every transplantation or any methodology, what we are doing is like many places they are following many methods. Those methods can be taken as like a key key for the transplantation, and it can be modified depend on the places or water quality or the sediment quality at the places. And for Gulf of Kutch area, we found this methodology is suitable. And so maybe in other parts we can go with the like some modification of this methodology. And and thank you for this for listening. Thank you very much.
Thank you, sir.
Thank you very much, sir.
There are a few questions.
Yes, sir.
What is the medicinal value of seagrass?
Sir, actually now the medicine value few works are going on. The total marine organism itself, they have some medicinal value. So till now, many this medicinal property of the seagrass is not that commercialized. Still, the research is going on, sir.
How many species of seagrasses are present in the Indian coastal water body?
Sir, nearly it is 16 species are found in the Indian coastal water. That is according to 2015 report, and after that there were very less reports.
There's one more question from how the microplastic content in the marine ecosystem influence the seagrass?
So actually the marine this microplastics in seagrass, again the associated organisms, they feed on the fine particles like the sediment flowing sediments. So this microplastic plastic along with this fine particles, they would enter the digestive system of the associated organism, because this associated organism also plays an important role in seagrass ecosystem services. Like the excess nutrients or excess coliforms and all are digested by this organisms which is associated with seagrass. When these organisms are affected or affected by the microplastic, then there will be there will be imbalance in the seagrass ecosystem.
This is something related to extinction. The part of marine organism lay eggs on the seagrass. When seagrasses are disappearing from the environment, what is the impact of these aquatic organisms?
Again, see, the problem is same seagrass is also it is always providing a habitat and breeding and all ground for many organisms. If the seagrass ecosystems are vanished, obviously these organisms also will be affected. That is the main reason what not only seagrass, all the ecosystem right from salt marsh till coral reef mangroves and some mudflat and seagrass ecosystem. If anything is affected, all the organisms will be affected.
The next question is, what is the role of dugongs in maintaining the seagrass ecosystem?
Sir, this dugong, they feed on the seagrass. They go for browsing the seagrass. They will not uproot the seagrass ecosystem. And once as we see in the terrestrial, if we prune the plants or seagrass and maintain the lawn, it will be growing. Something like pruning. So this dugong, their fecal matter and the way of feeding is also maintain the seagrass ecosystem, so that it will be in check, so that there will be no decomposition of the leaf happens or the reddening of the leaf from disease. So everything will be in check. So dugong has a very important role in the seagrass ecosystem.
The last question. Do you think it is a good idea to declare a place where there's an abundant amount of seagrass as MPA in India? At least one place.
So there is there are many places with rich biodiversity. According to in India, like if we have like a mosaic of habitat, including seagrass or coral and other ecosystem are declared as marine national park and in Andaman, Gulf of Mannar and Gulf of Kutch area. And the other places like many places which is outside the marine national park also have a seagrass ecosystem, but considering the livelihood of the fisher folk, they are not declared as a ecosystem in marine national park. Rather, declaring marine national park because marine national park the fishing activities and livelihood will be limited, so that the local community will not be able to cope up with their for their livelihood. So they can be trained or they can be educated or create awareness about the seagrass or how to use like fishing gear or anchoring in the seagrass ecosystem. Because even in Park Bay area and Mandapam and the Tangasimadan area, there are a lot of seagrass which is outside the protected area. And again in Gujarat, there are many seagrass ecosystems are out of the protected area. And but still it has to be conserved. Like the main thing is like the fisherman community, if they are trained or if they have awareness about this, they they are the first people who are using the marine ecosystem. So they should be like educated on more on this. Seagrass.
I think just we'll finish up with one more question. So this is a question from the YouTube participants. The phantom depth of the growth of the seagrass. The depth of the growth
Okay.
of
So like depth is that is very depends on the water quality. Like what happens like this seagrass they depend on the sunlight. So like they grow till where the sunlight reaches. In Japan, they have even recorded nearly figures at 30 meters. So again, it is based on the like sunlight where it reaches.
Thank you, sir, for the clear explanation. You have answered all the participants' questions. I think a lot more questions are there. Because of time constraint, we are just concluding now. Over to Dr. Tirunavakar, sir.
Thank you, sir.
Yeah, first of all, I would like to thank Dr. Adavan having accepting our invitation to deliver lecture on seagrass and its conservation. He has explained the seagrass diversity and species, the restoration of seagrass, especially the case study conducted at Gulf of Kutch, Gujarat, and the conservation aspects. Thank you, Adavan, once again having given the lecture. Thank you.
Thank you very much, sir.
Thank you, Advan, sir.
Thank you, sir.
Dear participants, now we move on to the second speaker of the day, Dr. Titus Emmanuel, Research Assistant, Marine Biological Research Center, Zoological Survey of India, Chennai. I now request Dr. K. Samiappan, Assistant Professor of Zoology, Dr. Ambedkar Government Arts College, to introduce our resource person, Dr. Titus Emmanuel. He is going to present on the marine sponges and corals.
Morning, one and all. I express my sincere thanks to our HOD and faculty members for giving this opportunity for me. Today's session on National Capacity Building Program is deal with the marine biodiversity. I am here to introduce the resource person for this session, Dr. Titus Emmanuel, Research Assistant, Zoological Survey of India in Lakshadweep Island. He did his bachelor degree at Madras Christian College, Chennai. He did his master and his doctoral. He has did his PhD at Annamala University, Chidambaram. His thesis presented is diversity and distribution of marine desmospongiae in an Andaman and Nicobar Island. And he served as assistant professor, Department of Zoology, Madras Christian College for 1 year. He has been part of the various research projects in Andaman and Nicobar Island, dealing with coral reef monitoring and the effect of herbivory in coral reefs. He worked in Andaman Nicobar Island as a researcher in Zoological Survey of India, ICR, Pondicherry University. His area of interest in augmentation, biodiversity in remote island ecosystem. He has contributed 2 books and 18 research articles published in Indian and international journals. Next, healthy marine ecosystems are important for society's science. They provide services including food security, feed for livestock, raw material for medicine, building materials from coral rocks and sand. Now he will proceed with his presentation on marine biodiversity. Thank you, sir.
Thank you, Dr. Samyapan, sir. I now request Dr. Titus Samuel to share the screen.
Thank you, sir. Thank you for your kind introduction.
Yes, sir, your screen is visible.
Yeah. Okay, so thank you one and all for joining us today, and thank you for the organizers for giving me a chance to expose the beauty and biodiversity of marine sponges and coral reefs and the need for their conservation. So the scheme of the presentation is I'll be introducing the sponges and a little bit of taxonomy of Demospongia, which was an important part of my PhD, the biodiversity studies that have been done in India, the ecological significance of sponges. Then I would move on to corals, the threat to coral reefs, significance of coral reefs, and the conservation of coral reefs. Of course, the sponges that I've studied are also mainly from coral reef environments. So I thought putting both of them together would be more interesting and would actually cover the topic well. Okay, so sponges are a basic introduction. They are very primitive organisms. They are the first of the multicellular metazoans. As life evolved, these cells came together and started to function as a single organism, and they lack any tissue grade of organization. They are sessile organisms, and they are filter feeders. So here you see a very basic structure of a sponge. They are much more complex in reality, but this is a very basic structure. So if you can see, there are different types of cells. So there are collar cells, which are the very important functional cells, which are on the inner layer of the sponge. They do a lot of they have a flagellum which beats and causes a current that causes water to enter through the ostia, which are the smaller pores on the sides of the sponge, which enter into the spongocele, which is the cavity in the sponge, after which it goes out through the osculum. During this process, these collar cells, which have a collar or a circular disc of cilia, trap food particles, bacteria, and small even chemicals from the water, and they ingest them, and through that they pass them on to something called amoebocytes, which transport these nutrients to other parts of the cells of the body. So here you have a little detailed view of the choanocyte chambers. The choanocytes are the collar cells, and how these water currents move from one place to the other, and how they enter into the sponge cavity. So during this process, the nutrients are absorbed. So here, a small GIF to show how fast the sponges, are able to filter water. So the dye that is used is called fluorescein, which is a non-toxic dye. These have been used to study how fast sponges can, filter water through their body. Okay, going on to classification, sponges are classified under the phylum Porifera. So Porifera meaning pore bearing. So you saw that they have lots of pores, ostia, osculum, and Demospongia are the major group of sponges which accounts for about 85% of the sponges. Calcarea about 5%. Hexactinellida, which are deep water glass sponges, which are about 8%. Then newly erected Homoscleromorpha, which is initially under Demospongia, also accounts for just about a very little 2%, but we really don't know much. We might find many more of these sponges. Then, so during my PhD, I dealt with mostly on Demospongia, and Demosponges are the most common in the coral reef environment, and they are also very common in all areas of the ocean. So, for example, you can see that more than 7,500 species have been reported on the Demospongia. The total sponges are about 8,500 species. So you can see how much percentage of sponges are under this group. They are mainly marine, except about 150 species, which are considered a different order called Spongellida. Except that form, which are freshwater sponges, all the others are marine sponges. The skeletal makeup is made up of either siliceous spicules or spongial fibers, or both put together. The diversity is, like I told you already, is highest at the shallow tropical environments, especially in coral reefs. And they play, in some areas, in some coral reefs, they are more diverse than even the corals themselves. Corals in the coral reefs are very diverse, but sponges in some of the reefs have been seen to, even beat their diversity. Okay, then they are classified into three groups: Heteroscleromorpha, Keratosa, and Verongimorpha. So in Heteroscleromorpha, you can see the sclerites, or the spicules as they are more commonly known, are very diverse. So there are different types of spicules that are made up of silica. So these siliceous spicules make up the bulk of the skeleton, and these are also supported by spongin fibers in some cases. In Keratosa, these spicules are amiss. They are not found in Keratosa. These Keratosa are called Keratosa because they are, keratin, which is a protein. These spongin fibers are also very similar in protein. They are proteinaceous in origin, and they form the skeletal structure mainly. And then there's the subclass Verongimorpha, which do not have either of these. Sometimes they might have siliceous spicules, but they do not possess spongin fibers. They are mainly made up of collagen, and that is their body makeup, and they're very cartilaginous to feel. So these are some examples. And so sponges, how they are classified, a little bit about how the main basis of classification. So siliceous spicules, so the chemical makeup is important. Siliceous spicules are Demospongia, Hexactinellida. Calcareous spicules only in Calcarea. Spongin fibers are seen only in Demospongia. So based on these, we can. The major thing that we know is the structure of the skeleton. So the structure of the shape and the different forms, they are so diverse. It is amazing to see such, sometimes they're very uncharismatic to see from the outside, but inside, when you extract the spicules and see, they have so much beauty, and there is so much intricacy in the spicule structures. So see, these are some spicule structures, and followed by spongin fibers as well. And then you can see the arrangement and orientation of the spicules are also very important. How they are arranged. Sometimes they're radially arranged. They're arranged from the center towards the outside. Sometimes they are just a net, like the last example there. So these are all important. You should classify. Then the next is the dimension and the size. So the spicules are classified into megascleres and microscleres for basic understanding. And the megascleres play a very important role in the structural integrity of the sponge. They have very clear, they form clear structures. The microscleres, not very clear understanding is there about why they are present to give structural support, but very rarely they are known. People think they are there to see they have a lot of spines and all that, so they might be to deter predators from feeding on them and so on. So for example, some of them have a cortex of these microscleres, so they mainly defend against nudibranchs that feed on them. So that might be the main reason for this. So now I'm going to show you some examples of sponges that I have identified from the Andaman Nicobar Islands. So through my PhD study, I was able to record about 103 sponges from the Andaman Nicobar Islands, and also to document their similarity between the island groups. For example, from Andaman Nicobar Islands, is a very long island group. So there's Nicobar group of islands, which is much down south, and you have the South Andaman, North Andaman, and Middle Andaman up north. And there were a lot of differences between the islands, and they also showed similarity between the African species, the Indo-Pacific species. All that was recorded through my study. So these are some examples of the sponges. And so on top is the in situ image. Below I have some, the skeletal structures and how they are arranged, and then the spicules at the bottom. So these are again some more examples. I'm going to run through them a little fast, but as and when I find something interesting, I'll explain a little more. For example, this one is a Clyonid sponge. Clyonidae is probably very common in our textbooks. These are boring sponges. They excavate into calcareous structures, mainly coral rocks, and they dissolve the coral calcium carbonate and give it back into the environment. So that is they play a very important role in getting the calcium back into water. So again, very interesting spicule shapes. So that's why I have shown you these. So here the spicules are supported by spongin fibers, which are clearly visible here. Again, this sponge is very interesting. It always grows along with a algae. So they can't be separated. They are like one organism. There is an algae and the sponge. They're growing together. So the surface layer, you can see the spicules. Below that, the brown layers are the algal component. These are very charismatic species that are known all over the world. These are called barrel sponges. This Zestospongia testudinaria, the Indo-Pacific species. This is very big. It can be up to 2 meters to 3 meters long or wide, and it can fit a human being inside their barrel. And these play a huge role in increasing the structural complexity of the reef itself. And it harbors sea cucumbers on them. Fishes go and take refuge. So many other things. So they also are an integral part of the coral reef. So this is a very interesting pink ball sponge. The ball on top is very interesting in that it is used as a propagule by the sponge. Not very commonly observed. It releases this ball, and wherever it settles, it can form a new colony. So it's kind of a bud. So again, very interesting spicule shapes. That's the reason I've added these. So now you can see these sponges come under keratosa, they do not possess any spicules. So they are mainly spongin fibers, and the spongin fibers here are in the form of a dendritic skeleton, dendritic meaning tree-like. So you can see how the skeleton is. Following which, these are anastomosing spongin fibers, which form networks, and they also do not possess spicules. So these come under Verongimorpha. This is one interesting group where the Verongimorpha also have some, they developed spicules as well. So they have asters, but they don't have any other kind of spicules. So these do not have spicules, neither spongin fibers. Very interesting species. Looks a lot of times these can be even mistaken for tunicates. Okay, so the sponge taxonomy or the biodiversity studies started in 1794. The first sponge that was described, it was from Tuticorin. After that, about 60 years later, Bowbank and Holsworth, they described about 4 sponges. Then the actual starting came when Carter in 1880 he started describing species. About 54 sponges he described. This I'm going to just run through quickly. So the beginning schools, these are all the beginning times of sponge taxonomy. Then Carter period is what is highlighted right now. Then it was followed by schools again, and then this region was mainly by Carter and Dendy. So Carter trained Dendy, so that is very important. I just wanted to show how mentorship is very important in biodiversity studies. And very similarly here, Burton, another scientist called Burton, was trained by Dendy, followed by here P.A. Thomas, who was trained by Rao and people, and then there was a lull in between in this time, because there were not many people after P.A. Thomas who actually took up sponge taxonomy. But in the recent times, due to their the chemicals that we get from sponges, there is a lot more interest, and people have taken up sponge taxonomy and biodiversity studies. Okay, so the ecological significance of sponges. Sponges play a very important role in the coral reefs, and they do a lot of filtration of water. So some species can filter up to 20,000 liters of seawater per kilogram of tissue. Okay, this tissue is not the wet weight, it's the dry weight, but still it is a really large amount of water that is filtered. This is more on the higher range. Some of them even filter up just about 1,000 liters of seawater per kilogram per day, but that is a huge amount of water that is filtered. So when they filter, they remove sometimes up to 95% of bacteria from the water, and some are consumed, some are used in their body as symbiotic bacteria as well. Sponges are known to house plethora of bacteria in them, and so they filter and convert dissolved organic carbon, suspended particles, and dissolved matter into food, and these in turn is converted into particulate organic matter for other organisms to feed. So sponges not only feed are extremely good filter feeders, they also help in other organisms to feed by making particulate organic matter that is expelled out of their body. Then sponges are thought to also play a role in the recirculation of carbon, silicon, and nitrogen. So they also play a huge role in benthic and pelagic coupling, which is the exchange of nutrients from the pelagic region where water and air interact, and the benthic region where there is much less interaction, but because of the filtering they do, they connect these two ecosystems, and the particulate organic matter is supplied to the benthic organisms as well. So They are great micro habitats. They have, they house lots of organisms like shrimps, crabs, molluscans, and algae, and so many other things, and bacteria. In fact, they are called holobionts. They themselves act as a niche in themselves. Food, they are also a source of food for a lot of sponge eating fishes, nudibranchs, especially some sea turtles, especially the hawksbill sea turtle is known to feed specifically on sponges. Okay, then let's move on to corals. So corals are, they need hard substrates to settle on. They are found in shallow waters. They are found in the continental shelves in warm climates. So the distribution is mainly within 32 degrees north to 30 degrees south latitudes. The temperature range is 18 to 36 degrees Celsius, whereas the ideal is 25 to 29 degrees Celsius. The depth seems to reach up to 90 meters. And even recently, they have found till 200 meters. One group called the Agaricidae are able to live in really deep waters without sunlight. But their ideal range is from 0 to 20 meters. In tidal areas to about 20 meters. The salinity is 27 to 40 ppt, but the ideal ppt is 35 ppt. pH 7 to 8.4, ideal 7.8 to 8.2. This is the they prefer this kind of thing. And moving on, the general structure. So the sedentary cnidarians with calcareous, horny, or soft skeleton. Okay, these most corals are colonial, and except some sponges like the mushroom corals, they rely on photosynthetic algae in their tissues to obtain energy from the sunlight. So these photosynthetic algae are called zooxanthellae. We'll look a little bit more on them as we go. So now we are concentrating mainly on hard corals, or the scleractinians, which are reef building corals. These secrete calcium carbonate outside their body and form a skeleton, which is the primary component of the coral reefs. They're generally distributed in the shallow, warm, and tropical seas, as we all know. Then a little bit on their feeding and nutrition. They all have stinging cells. Most of them are reef building corals, and they contain zooxanthellae or symbiodium species. Here we see some, an example of the zooxanthellae. This symbiodium species is very important for corals, as they require this energy. They can feed from using the tentacles, but they are not successful enough to survive only based on them. So they have, evolved to have a symbiotic relationship with Symbiodium, which provides the major nutrients to corals. So, okay, this is a very critical relationship, and you have to remember this. So the presence of so corals can be classified based on the presence of zooxanthellae. There are some corals like Tubastria, which do not require zooxanthellae for their survival, but they have very well equipped apparatus to capture prey. So they are called azooxanthellate, and the more commonly what we see is zooxanthellate corals. So then, based on the reef building ability, they are classified into hermatypic corals or reef builders, and ahermatypic corals, which do not play much of a role in building corals. For example, a solitary coral like the Parasites here shown, they're very small corals, and the soft corals also, they don't play a major role in the reef building. Okay, so coral reefs house a huge amount of biodiversity. So they are the greatest, they house the greatest biodiversity of all marine ecosystems. And if you can see, the reef fauna include the maximum number of phylums in any single ecosystems, including terrestrial. So the highest biodiversity that has been recorded is in the tropical evergreen forests. But the tropical evergreen forests have a lot of insects, which is of one class, Insecta, which increase the biodiversity by a lot. If that is not considered, the coral reefs would be the most biodiverse of them all. If you can see here, there are about 22 phylums that are found in the coral reef ecosystems. I'm sure there will be many more as well. Okay, so corals generally, how they are studied? They are studied in the, of course, they have classification, that classical taxonomy and all, but we study them based on their growth forms as well. So, for example, branching corals, they have a lot of structural complexity. They have, organisms can go between the branches and rest. So that you can see there are submassive forms, massive forms, which are more spherical and large, encrusting forms, which are just growing over the coral, over other substrates, mushrooms, which are solitary corals, and they are free. Foliose corals, which form leaf-like or plate-like, protrusions. So here you have an example. The first one is a tubular coral. The one on the right is branching coral, and then you have submassive forms at the left-hand bottom, and you have a massive form on the right-hand bottom. So these all make up a reef. So you have an encrusting form here, solitary form, and foliose form. So all of these are an integral part of the coral reef ecosystem to increase the structural complexity. So we cannot say one of these growth forms is more important than the other, but they all play a very important role. For example, the branching corals are very, very fast growing, but they are also the most prone to, being affected by climatic changes. For example, bleaching affects branching corals first, but the porritus or the massive corals have a lot of resilience. They are not easily affected by these changes, but they do not house the same amount of biodiversity that the branching corals do. So that is how they are. They are all important in their own way. Okay, some threats to coral reefs. So I've just classified them under natural and anthropogenic. Sometimes they are a compound effect of both. So the crown of thorns starfish, which has been seen to suddenly increase due to the climatic changes right now, increase in water temperature. Their survival of their larval stages have become better, and therefore these crown of thorns, which are corallivorous, they kill a lot of newly formed small budding corals. So when they are in huge numbers and there is a bloom, they can be very detrimental. They are part of the ecosystem, but when they are found in excess numbers, they can be very detrimental. Polychaete worms that the Christmas tree worms and the feather stars, they all bore into corals, making their structural integrity bad, and thereby affecting the corals. Bleaching and diseases, I will deal with them much more in detail, so I'll go next. Then tsunamis, earthquakes, these are rare but very, very destructive things to the coral reefs, because they can damage huge areas in one go in a day. And so, but coral reefs can, bounce back from these as well. Sedimentation, though naturally also they occur, but they are increased due to human activity. And wherever there is lots of human activity, there is a lot more sedimentation. And as you all know, corals require less turbid areas to survive. If you can see the survival of corals in coasts where there is a lot of runoff is very rare. Only in places like the Andaman Islands or the Lakshadweep Islands do we see lots of corals where there is no runoff and less turbidity. So sedimentation is also a bad thing for corals. So diseases caused by our inclusion of sewage and other things is also a major problem recently. Then unregulated tourism. So there is a lot of problems when there is a tourism which is not which is only focused on making money, but there is no regulation based on how they have to do. So there is a good way and proper way of doing tourism, and thankfully people are nowadays being trained, and hopefully that will also increase make the coral recovery better. Overfishing and ghost fishing and leaving nets on coral reefs, all that can also be detrimental. Souvenir collection is very common. People like to have corals in their homes. They want to they're beautiful, so they want to adorn their shelves with them. But more with the trade restrictions, they will be reduced. So coastal industries letting their toxic waste to run off into coral reefs also can be detrimental, and shipping with all its oil spills and all that is also very detrimental to coral reefs. So these, for this, the crown of thorns, you can see some the from overfishing or bad fishing practices. Here you see the polychaete worms, the Christmas tree polychaetes, which have infested a coral. And here a bad practice of which damages the corals in the long run. Okay, coral bleaching. So I told you this is a natural threat. Coral bleaching is when the zooxanthellae that are present inside the corals are are forced to leave their host due to the abnormal increase in water temperature. This causes the coral to look stark white in appearance, because the zooxanthellae is what gives the major color to the corals. So when this happens, corals are not able to survive for too long unless the zooxanthellae settle back. If these stressors, that mainly the increase in temperature, goes away soon, the bleach coral can again house the algae and get back to normal. But generally, due to global warming and the climate change, and recent years have been seeing much more prolonged summers and much more intense high temperature ones, which is also coupled with El Nino effects. So what happens is these corals are subject to these adverse conditions for a very long time, and these bleached corals often die. So this is a major problem to coral reefs and their survival. So this is man-made, the in the sense of climate change being because of human beings and the greenhouse gases that we have been introducing into the environment. It is considered a natural thing because it happens in the natural ecosystem itself. Okay, so then this is coral diseases. Coral diseases have been there. They have not been so prevalent till the last decade. So recently studies have been documenting all of these diseases and how much they affect the coral reefs, and their growth rates, the community structure, species diversity, and all of these are being affected by coral diseases. And as the climate change becomes worse, the coral diseases also seem to be becoming worse, and humans, by letting out us sewage, and because of eutrophication, we are just making things worse for the coral reefs. Okay, why are coral reefs so important and some significance of coral reefs? They protect our coastlines from tsunamis, from harsh ocean storms and floods. Ocean storms sometimes are never felt in the coastal regions, but they are felt in the sea, and you can see there are a lot of damages in the coral reef environment, but they shield the coastal villages and the coastal towns from the adverse storms. So then coral reefs support a variety of commercial and artisanal fisheries, and they need not be only in the coral reefs, but because they function as breeding and feeding grounds, these fishes or these fisheries move from here into the coral reefs and then move away, and thus contributing to a larger area of fisheries. Then of course tourism is a very important part of coral reefs, and all over the world more and more people are getting into scuba diving and wanting to be adventurous and see what is under the ocean, and this is a revenue for all coastal towns, and thereby increasing the economy of every country that has it. So also there's a lot of coral organisms or reef inhabitants that have different types of chemicals that are being found, novel chemicals that are being found, that are anti-carcinogenic, they can be antihistamine, they can be painkillers, they are anti-inflammatory. They have so many different uses. Some are used for other things like they reduce corrosion in the ship by being not letting attachment of fouling organisms. So anti-biofouling is what I wanted to say. And so these uses are found, these chemicals are found in organisms that are not just corals. For example, sponges are very, very important part of from where organisms from where chemicals have been derived. So recently I read an article which said about 60% of the new chemicals that are being extracted from the ocean are from sponges. And sponges, if you can see, they are not very intimidating. They don't have much defense structures. So over years they've evolved to produce different types of chemicals that deter predators to prey on them. So these chemicals are their primary defense mechanism, and so these chemicals can be used by us for various other things, and thus the sponges also play a very important role in for the humankind. Okay, a little bit coral reefs, reef resilience. So reefs are very resilient, but due to climate change, they're also we are losing several reefs all over the world. So reefs are very interconnected with the other organisms. Corals are not just corals, or you've seen there are a lot of biodiversity in the coral reefs. So herbivory is a very important part of coral reefs. The fishes, they feed on algae, making sure that the corals have space to settle. The coral larvae have space to settle. There is something called crustose coralline algae, or CCA, which needs to be present for chemical cues for the larvae to settle. So this is also provided by the herbivory. When herbivory, the macroalgae are consumed by herbivorous fishes, and the tuft algae are being consumed by herbivorous fishes, the CCA underneath is exposed, and there is space for corals to settle, the coral larvae to settle. One other very important part is that the top consumers need to be present. For example, sharks around the world are being depleted. Especially India has recently been declared one of the most depleted stocks of sharks, and this is a major problem for us, because these top consumers do not feed on they do not feed on big piscivores like groupers, which in turn increase in great numbers and feed on the herbivorous fishes, and the herbivory is completely reduced in some reefs, thereby reducing the reef resilience. So reef resilience is a compounded effect of all of these things. Okay, so the conservation strategies, I think I'm running out of time, so I'm going to go through it quite quickly. So there are different types of strategies, either directly protecting particular animals, like for example sharks, which is very important, but in some other cases where we need to protect the ecologically significant areas by declaring them as protected areas. So like Adavan had already told you, there are areas that are protected for seagrasses as well as coral reefs, and this helps in protecting a stock, or an area which is not being affected by humankind, so that the other reefs that are connected to them, which may not be protected, will also benefit from them by way of the biodiversity from here moving to the other places. So this connectivity is very important, and that is a major point why biodiversity needs to be studied, and we need to know what kind of organisms are present to know which are the areas we need to protect. Okay, then there are active conservation strategies, like for example restoration of reefs, where we do coral gardening, and setting up underwater frames to aid in coral settlement, and to increase this increases structural complexity. Artificial reefs have been constructed by sinking ships and allowing corals to settle on hard substrates. For constructing artificial reefs to serve as a barrier in some cases, and also in some cases to act as FADs. So these are some of the structures that are seen. So this is a coral frame where corals are attached, and this is allowed to grow, and this increases structural complexity. It acts as an FAD. Fishes come and aggregate over there, and this is one way of actively conserving coral reefs. So on your right, you see a structure called a reef ball, which is a concrete structure which is thrown into the ocean, and this aids in corals to settle on them. And here you, on the left bottom, you see a shipwreck, or it might be a shipwreck or a ship that was submerged for the purpose of being an artificial reef. And these are some of the things that people are doing to, actively increase the coral reef areas in our area. The depleted reefs are also supported by these structures. Okay, thank you, and thank you, sir, for giving me the time. Yeah, I'm done.
Thank you, sir, for that wonderful and colorful presentation of yours. There are a few questions, sir. Do marine sponges affect the ecosystem of aquatic organisms? Do marine
Yes. So they most definitely do. So, for example, there are large sponges like the barrel sponge that I told you, that they house specific echinoderms, and they house specific fishes in them. So they do affect, and the climate change also affects these corals. In fact, they also undergo bleaching. Very similarly, they also house zooxanthellae in them. So sponges can also be affected by the climate change, and them not being there would affect organisms that depend on them. So it definitely does affect the ecosystem.
The next question is, what are the basic differences between a marine sponge and a freshwater sponge?
This is more of a taxonomy thing. They have very different spicules. The intricacy of spicules in freshwater sponges are very different, and they also have something called a propagule. A propagule that is covered by a very specific type of spicule, and so this is not observed in any of the marine sponges. So this particular thing will be enough to differentiate a freshwater sponge from a marine sponge.
How are sponges affected by pH change and global warming? What can be the mitigation process?
Did you say sponges or corals?
Sponges, sir.
Okay, so sponges are affected by ocean acidification is affecting a lot of organisms, especially more than sponges. They are affecting coral reefs, and coral reefs are, in essence, the ecosystem where these sponges are. So it's not like these are not existing in they both exist together, and when the ecosystem or the habitat in which the sponges are not in a proper state, the sponges get affected also. See, sometimes the sponges overgrow corals, but that is only for some time. After that, after it reaches a particular point, they again all die, because they cannot support without the corals also supporting them in the equal in an equilibrium. Yeah.
The next question is, what are the characteristics of homoscleromorpha that made it different from demospongia?
Okay, the homoscleromorpha have very small spicules, and the spicule structures are not quite different. The main way it is differentiated is by the way the spicule is secreted. So there are called two cells called sclerocytes, which are important for the spicule formation, and the way they form the spicules in demosponge. The other one, that is heteroscleromorpha or demospongia, is very different from how it is made in homoscleromorpha. But one more thing is, they are very, very small spicules. They are often considered as only microscleres, and they possess only microscleres. They do not possess megascleres. Yeah.
Another question is, what are the major fish species that live near the sponges? That is.
Yeah, there are so many different species: butterfly fishes, hawk fishes. Hawk fishes, you can go search the family Cirriptidae. So they uh, sit on sponges, and then they are, you know, predators, ambush predators which sit on sponges. There are other small blennies that live inside cavities of sponges. There, there are a lot more crustaceans that are directly dependent on sponges, which are excavating inside sponge colonies itself. So, for example, there are pistol shrimps, which are known to form ant-like colonies where there is a queen, and then there are a lot of other workers and stuff like that. They live inside sponges. So that also is there.
The last question, sir.
Yes.
How do sponges help coral reefs thrive in the ocean? Is there any interlink between them?
Yes, for like I told you, the water in the coral reef is constantly filtered by sponges. So the water quality that coral reefs, depend on, is being given because of the sponges that are present. They do it very quietly, and they're not charismatic, so people don't realize what a huge role they play. But they are filtering, like I told you, about 20, some species filter 20,000 liters per kg of their body weight uh, every day. So this keeps the water quality in the coral reef environment good. Also, by converting dissolved material into particulate organic matter, they supply food for a lot of other organisms that live in the coral reef environment.
There's one question from the YouTube. So what is the role of artificial coral reefs in the marine biodiversity conservation along Tamil Nadu coastal areas?
Okay, so there have been several projects that are being done. So one particular project, I think in the Tamil Nadu coast, I'm not very sure who is doing it, but they are using the very similar structures like the reef balls, but they are using triangular structures, and they are placing it in areas close to reefs and close to coastlines. So some of them function as fish aggregating devices. So they don't exactly house corals on them, but they themselves play the role of a reef. And these uh, increase the amount of fishes that aggregate there, and local communities are enhanced by they enhance their fishing in those areas. And also, in some areas, when they are placed near the reef, corals settle on them, and they form coral reefs, artificial coral reefs themselves.
How to differentiate dead and live corals?
So bleached corals are look dead, but if you look closely for a diver, you will know that when you go close to the thing, you can see life. But as soon as it dies in a reef, the first thing that happens within 24 hours is algae will settle on them. The white bleached coral is generally alive. Then after that dies, algae will settle on them. So uh, uh, algae are like tufts, so they'll be like hairy on top of them. So with that, you can find out that they are dead coral. Otherwise, corals have different colors. Some can be drab brown to green, bright orange, bright green. So different colors are there. So colorful ones are alive. The more the dead ones are usually white or covered with algae.
Thank you, sir, for the clear explanation. Over to Dr. Tirunavakar, sir.
Thank you so much, sir.
I would like to thank Dr. Titus Emmanuel having accepting our invitation to deliver a lecture on marine sponges and coral reefs and their conservation. So he has very clearly explained the sponge taxonomy, particularly the electron microscopic pictures of spicules. Further, Andaman and Nicobar Islands diversity, nearly 1 or 2 species he has identified, and the genesis of taxonomy of sponges regarding to the sponge side, and further corals, factors affecting corals inclusive of bleaching and diseases. Further, the conservation aspects and the economy, how it behinds the coral reef ecosystem. So all the aspects he has covered. Very wonderful presentation and nice slides. Thank you, Titus. Thanks, sir. Thank you.
Thank you, sir. Dear participants, now we move on to the third session. We have Dr. M. Arumugam, Associate Professor, Faculty of Marine Sciences, Annamala University, Chidambaram, Parangipete. He is going to be present on marine mollusk. I now request Dr. M. Sashikala, Assistant Professor of Zoology, Dr. Ambedkar Government Arts College, to introduce our resource person, Dr. M. Arumugam. Over to Shashikala, ma'am.
Good morning to one and all present here. It's my immense pleasure to welcome and introduce our resource person, Dr. Arumugam Muthuvel, Associate Professor in Center of Advanced Science Study in Marine Biology, Annamala University. On behalf of Zoology Department, Dr. Ambedkar Government Arts College, Chennai, welcome, sir. An eminent person with a wide knowledge of research, dedicated teacher, and 16 years of teaching experience. Dr. Arumugam sir specialized area marine biotechnology and marine natural product. Under sir guidance, 6 students awarded MPhil degree and 6 students awarded PhD and 4 students are pursuing PhD. Sir contribution on both major and minor project sponsored by UGC, YSAT, MES, DBT and DST. A dynamic person, sir secured a plenty of awards and certificates in research field. Let me mention a few. Tamil Nadu State Council Science and Technology Student Fellow in 1999 for involving in drug discovery project. Senior Research Fellow in 2001 for collection of documentation of molluscan resources at Andaman Nicobar Island. DAAD Summer Trainee Award from Leibniz University of Germany. DST Travel Grant Award in 2011. NUS Visiting Fellow from National University of Singapore in 2012. In 2015, SVA GATA Fellow from KTH Royal Institute of Technology in Sweden. And Best Researcher Award in 2016 and 17 from Annamala University. To his credit, 64 research papers have been published in peer-reviewed, reputed international journals and acclaimed for citation of his unique paper published by research paternity throughout the country in marine science. Warm greetings, sir. Now I call upon Dr. Arumugam, sir, deliver his valuable presentation. Welcome, sir. Thank you.
I now request Dr. Arumugam, sir, to share the screen.
Thank you, Dr. M. Sasikala, for your wonderful introductions about me, and most respected the organizers for this wonderful webinar on National Capacity Building Program and Marine Biodiversity, and the authorities of Dr. Ambedkar Government Arts and Science College, Chennai. And actually today my presentation is on marine mollusks, that means their diversity as well as the conservation. And my dear friends, and we are all in the life science background, so that's definitely you must know what is the importance of the marine mollusk, and also what is that the importance in terms of their useful such as their food or medicines or other value-added products. So in this particular sessions, I will discuss briefly discuss about the marine biodiversity and followed by the marine mollusks, their conservations as well as their diversity. And just a minute, I will share my screen for you. One minute, friend.
Yes, sir, your screen is visible, sir.
Yeah, fine, thank you. So my title is Marine Mollusk and Marine Mollusk Diversity and Their Conservations. Before starting my presentation, Already the sufficient introductions given by Dr. M. Sasikala. In addition to that, who I am. And actually, I'm working as associate professor in Center of Advanced Study in Marine Biology, and I'm the University Faculty of Marine Sciences. And this is my center. And some of the recognition of our center, as you might know very well about what is the Center of Advanced Study in Marine Biology and their role in scientific community in national as well as the international level. And these are some of the recognitions from our the government of India side. Actually, the Center of Advanced Study in Marine Biology is a very premier center for marine sciences in India, and I am from 1957. And also, this is the only one center for Center of Advanced Study in Marine Biology since 1963. And what are the other recognition by government of India is, so the Ministry of Earth Sciences identify our center as Center of Excellence in Ornamental Fish Culture from 2004. And also the Department of Biotechnology identified our center as Center of Excellence and Innovation Biotechnology since 2010. And also the University Grant Commissions identified our center as center with potential for excellence in particular areas, especially in marine biology since 2011. And also the Ministry of Human Resource and Development identified our center as Center of Excellence in Frontier Area of Science and Technology from 2013. And also the Department of Science and Technology identified our center for promotion of university research and scientific excellence. And my dear friends, and each and every center definitely they have the vision and mission. And these are the vision as well as the mission for our center. And what is the vision? To understand the scientific principles of marine life and the environment towards developing a world-class institute in marine sciences. And what are the mission? Physical, chemical, and biological assessment of marine environments, biodiversity and biological studies of marine organisms, species recovery for the marine species or threats, restoration of the coastal ecosystem, marine bioprospecting for food, fuel, medicines, and other high value-added products. And also the mass production of marine organisms for sustainable utilization. And we are also involving training and capacity building in marine sciences. My dear friends, because this is the time for most of us, we are very much affected by the pandemic corona. And I hope that some of the parents as well as the students or the young minds watching this webinar. So this is a chance to share what are the programs we are offered by Center of Advanced Study in Marine Biology. We are offering the PG as well as the UG program. First one is the MSc Marine Biology and Oceanography program. It's a professional course that is first in India. And the next one is the Marine Biotechnology. It's a DBT sponsored program. And student selection is from the JNU. And also we are following the curriculum of DBT. And also we have the coastal aquaculture program at our premises. And also we have recently introduced the MFSC course, and the advertisement is waiting from our authorities. And also we have the two UG program at our centers, that is the bachelor degree of fishery science. It's another professional course for our center. And also we also introduced the BVOC, Bachelor Degree of Vocation in Aquaculture. In addition to that, we are offering the 5-year integrated program, that is the MSc Ocean Science and Technology. And at this introductions, I just to discuss come to the points. And regarding the biodiversity, we are very much known, and we are most of us, we are dealing or discussing day by day or year by year about the biodiversity. My dear friends, so what is that unique definitions for the biodiversity? So the biodiversity is defined as all hereditary based variations at all levels of organization, from the genes within a single local population to the species composing all or part of a local community, and finally to the communities themselves that compose the living parts of the multifarious ecosystem of the world. And in the case of biodiversity, we should concentrate not only the higher animals, we should concentrate from microbes to macros. And brief introductions about the coastal and marine biodiversity of India. My dear friends, India has a very vast coastline, about more than 8,000 kilometers. And also the exclusive economic zone, that is EEZ, of 2.02 million square kilometers. And also we have the coastal districts about 53. And also we have the maritime states about 10 and the union territories 6, including in Andaman Nicobar as well as the Lakshadweep group of islands. Nearly 25% of the country population resides in these areas. So that's why this the importance of marine biodiversity, and also most of our peoples which are residing or which are depends on the coastal ecosystem. And the coastal and marine biodiversity of India, because the oceans is just like a harbor or tremendous biological diversity. Almost 34 phyla are identified. Out of 34 phyla, 32 from marine environments and 13 are represented only in the oceans. Almost half, that means more than 50% are only live in the oceans. So that denotes the what is the role for oceans to accommodate all the organisms. The coastal and marine biodiversity of India, the potential of marine fishery resources in our country. As estimated, the potential yield is 3.93 million tons annually. In this case, marine mollusks contribute lots, especially from the bivalves and gastropods. They have yield or harvested almost 2.05 lakh tons per year annually. And then the deep sea finfish and other crustacean resources contribute 1.01 lakh tons. The production of marine products from the various marine sources comes 27.78 lakhs tons. Exporting the products, whether the value-added products or it may be the raw materials, which comes around 4.37 lakh tons. And also, the government of India earning 6,188.92 crores annually. And also the production of pelagic fishes more than 1.36 tons, according to the Mohan and Jayaprakash 2003. My dear friends, this the information shows how the oceans gives the valuable products or the valuable material from the marine sources. This is actually one of my friend Emmanuel just now engaging the coral reef, so that's why I don't engage the coral reef informations. But in my point of view, as a molluscan biologist, most of the molluscan species from 7 important classes, they are almost found in the intertidal areas, especially the coral reef areas. Because the total area of the coral reefs in India is estimated as more than 2,374 square kilometers. Because the coral reef ecosystem provides a good shelter for marine algal as well as the other diversified flora and fauna. So that's why the maximum diversity is not only for the algae or even the microbes. This is a place as a nursery ground to accommodate all the animals, all the faunal forms. So that's why the coral reef, the diversity is very much important when compared to others. This is the coastal marine biodiversity. Starting from, I already told you that the marine biodiversity, it is not only for the higher animals or the invertebrates, it including the microbes or the microalgae from the from microalgae to the macros, which including the diatom and dinoflagellates. This is the information for the what is that the percentage for the global productions or the global availability of diatoms as well as dinoflagellates. But Indian concepts almost 200 plus diatoms as well as the more than 90 plus Indian dinoflagellate species which are available on the intertidal regions of Indian exclusive economic zone, which almost coming 5.5%. And this is the coastal and marine biodiversity, starting from the other the faunal groups, starting from the porifera, cnidarians, ctenophora, and these are the other groups which are contribute much more in terms of the species diversity. as well as their percentage point of view. And this is a coastal marine biodiversity, and the other informations in from the other groups, and like the polychaetes, nematodes, they also contribute much more. And this is the another information from the Ketonota copepods. Some of the Ketonotas and copepods which are coming under the zooplanktons, they also contribute much more in terms of the diversity as well as the percentage point of view. And the other coastal marine biodiversity, which including the other groups such as the Ostracoda and the other forms, the Tardigrada, Lucifera, and another Cainorenca. So these are the species which are contribute almost starting from the 0.1 to 5% in Indian context. And this is the information for the other nematode swarms, which contribute almost 0.83%. At global level, more than 12,000 species are there, and Indian constants, it's almost less than 100. And this is information from the other important groups such as the crustacea, stomatopods, hermit crab, brachyuran crabs, and shrimps and prawns. And almost the global level, more than 150,000 species are available, and out of 150,000, 40,000 species are described. In Indian context, almost 2,934 species are available and contribute around 1.956%. And this information the phylum Mollusca, because the phylum Mollusca, this is the second largest phylum in the animal kingdom. The first one is the phylum Arthropods. You are very well known about the phylum Arthropoda because the insect groups contribute much more when compared to the other species. So that's why they give the first priority. And this is the second largest phylums. Almost globally, more than 1 lakh. Uh, 52,000, and my dear good friend Dr. Magesh Kumar, he will skip this information. And next one is this is the other Hermi chordata species. Globally, 100 species which are available. In Indian context, we have only 12%. So that's why almost we have the 12% in terms of their contributions. And this is a coastal marine biodiversity information for the fishes. Almost globally, 16,475 species which have been described. And Indian coastlines, the Indian context, we have 2,546 species, which contribute around 15.45. And this is the information for the reptiles, which including the sea turtles, crocodiles, sea snakes, like that. And globally, more than 120 species which are available. Out of 120 species, Indian context, 35 different species are available, which contribute 29.16%. And this is the other mammal groups of the mammal contributions, which including the dugongs, dolphins, whales. Globally, more than 500 the species are available. In Indian context, we have only 25 species which contribute around 5%, and which including these are the information for the marine birds. Then coming to my points, right? The marine molluscan diversity and conservations. This slide shows what are the species which have been collected just 6 months before at the Gulf of Mannar regions. Whenever we went for the collection, especially the Gulf of Mannar and other areas, and they have targeted the fishes for their fishing practices or through their fishing practices. But unfortunately, due to their untargeted fishing, they have exploits very valuable and very unique groups like the mollusk and other forms. So that's why it's a very interesting groups. We have concentrating or we are discussing about the second largest phylum, marine mollusk. And this is some of the brief introductions about the introductions point of view. Mollusks, the very major example is just like the slugs, snails, clams, octopus, squid, oysters, etc. And about more than 50,000 living mollusk species available, are about 35,000 fossil species are also available. And also the phylum Mollusca is the second largest phylum in the animal kingdom, I already told. The majority of the mollusks are marine, but large number of species which are also occupying the freshwater and terrestrial habitat. And some of the mollusks, they can swim, while others are sedentary. And most of the marine mollusks live as sedentary form. They can easily settle at the particular area, so then they can easily survive. So another information regarding what are the other unique features for the marine mollusks. My dear friends, you are very well known about the oysters, that means the pearls. Because the particular group of animals, they can producing the pearls, and through their recombinant technology, DNA technology, we have to developing what are the colored pearls we want. We can producing the colored pearls through their recombinant DNA technology, and we have produced different colors of pearls. And it's a very unique features which are present not only in the marine, it is also applicable in freshwater. And other common characteristics of the mollusk. Mollusks are soft-bodied animals. They are the soft-bodied animal. That means the very flexible body, and it having a bilateral symmetry, and some of the bilateral symmetry, and also some of them are act as the eucoelomates. That means they're having the body cavity, and the body cavity which consists of other internal organs, and also the digestive system, with two openings which are present, and just like a mouth in the head, the anus into the emptying the mantle cavity. And most of the animals, especially the bivalves, they're having a specialized teeth-like material or teeth-like projections. That is called as the radula. The radula usually a tooth. It may be a single or could be in thousands numbers of rows which are present. And it is adapted for scraping, tearing, stabbing, and cutting the food materials. So it's very unique features which are present on the marine mollusks. And in some of the teeth, they are hollow, and sometimes it may be poisoning or used as weapons. My dear friends, you know the conus. So this is the conus species, which are very potent. They have they can easily synthesize or secrete the very potent toxin. They can kill the human. So that's what the toxins. You can see the animal. This is very small animal, small size of animal, but they can producing a very potent toxins or venom which can kill the human. So their contributions are their unique features of the marine mollusks. They have to provide lot of information to us, because the learning is lifetime process. So we should learn from the small creatures like the molluscan forms. And also they are thick. Most of the mollusks are thick muscular foods, because they don't have any distinct food for their locomotor purposes. They're having a muscular food for their locomotions, especially in the gastropods. And some of the mantle, that is the thin membranes which are surrounding their coelom, and they can secrete the shells in the, especially for the shell process. The mantles only secreting the calcium carbonate and for developing or the individual shell. It's a well-developed head regions, well-developed head which may bear sensory tentacles. And most of the gastropods animals which are having a very sensory tentacles are present, except the bivalves. And also in the case of the circulatory system, except the cephalopods, cephalopods which are having the closed form of circulatory system, but they're having the heart, blood vessels, and the pigments is the hemocyanous. So that's why the blood is in bluish in color, not in the reddish one. And when oxygenated, such blood is bluish in color. When some of the deoxygenated blood is somewhat colorless. And even most of the molluscan form, they're having the pigments such as the what is that, cyanin, hemocyanin, but one group of bivalves, that is the, I think, one particular group, I just forget the name, and Anadara, I think, blood clamp. It is called as the blood clamp. They can producing the hemoglobin instead of hemocyanins. So there also, it's a very particular groups which are synthesis are producing the hemoglobin as a pigment for carrying the oxygen into their body or their respiratory purposes. And even most of the animals which are having the gaseous exchange, that means the respirations which are taken by a special type of gills, small rudimentary lungs, as well as some of the other body surfaces, for example, nudibranchs. And this is phylogenetic classifications of the phylum Mollusca. So I already told you that phylum Mollusca, which consists of or composing of 7 important classes, starting from the bivalvia, monoplecopora, gastropods, cephalopods, scaphopods, aplacoporans, and polyplacoporans. Out of 7, depends on the availability as well as the species richness. We have Consonantidae, and also almost the world which are concentrating only the bivalves, gastropods, and cephalopods. And these are some of the very small size bivalves to the larger size bivalves. So when we are watching this one, that is the Perna indica, and this is the Perla isis. So both the forms, they are somewhat small in size, but almost 4 to 5 feet bivalves is also there. That is called as the giant clam, because it's very huge size or huge level of clams are available. That is called as the giant clams. And this is the classes. I already told you that the classes of mollusks, 3 are its very major classes out of 7. Now we have discussed. In the case of gastropods, which including the snides, limpets, slugs, and conchs, very ideal example. This is called as the world name is the Xanthosperm, and the new name is Turbinella pyrum. And this is called as the sacred chunk or holy chunk. This is it's very much important, and most of the Hindus, they are preferring these shells, and they have worship their homes for giving the good health and wealth. And regarding the bivalves which composed of oysters, clams, scallops, because the bivalves, you know that the bi means as you know that the 2. So this is the bivalve species having the 2 walls. Having the 2 walls like this. This is the bivalves, the matrix. And these animals which consist of the opposite side, this same type of valves is present. So that is called as bivalvia, and also having the hinged teeth at the top. And this is somewhat the unique features for the bivalves groups. And in the case of cephalopods, as you are very well known, it's a very intelligent group of animals which are present in the animal kingdom, apart from the humans, if you have faith. And that means octopus, squid, and nautilus. These are the other major group of animals which are present coming in the cephalopods. And gastropods, under diversity, in terms of the diversity, gastropod is the largest and most diverse class in the phylum Mollusca. About 40,000 or almost 80%, which are contributing only from the class gastropods. And most of the animals have single or coiled-like structures. You can see these things, and I'll show one animal to you. And this is the just like a coil-like shells. This is a coil-like shells which are made up of the calcium carbonate, and the body can be withdrawn. The animals which are present on their inside of the shells, so they can withdraw when the animals are disturbed. They can easily withdraw their whole animals. That means the visceral mass. And shells are very pretty in color. You can see the things. They are very pretty in color. The species is Mitra species, Mitra, and Mitra strictica. 2 species, important species which are available, especially in India, and very particular in Great Nicobar Island. When we went to the Great Nicobar Island during 2001 and 2004. We have collected some of the very important features which are present at our laboratory. And also the about 3,027 species have been reported from India, from the gastropod groups belonging to more than 220 families and 571 genera. The slugs, that is called the shellless mollusks. What is the unique features of the mollusk? That is the shelled animal. But this is one group of animals which is also coming under the phylum Mollusca. But most of the mollusk animals, they are having the external shells. Outerly they have present. But in the case of the slugs, nudibranchs, the shells are very less. That means the rudimentary shells which are present of inside the animals. So that's what it's a rudimentary in nature. So when we are seeing such kind of the slugs, it's just like a shellless animals. It's ranging size starting from the very it's microscopic to the 24-inch slugs which are present. And its very ideal example is the sea hare, that is Aplysia. And another information is the feeding habitats. It's most of the signs that the gastropods animals, just like the herbivores, and sometimes some of the animals are the piscivores. For example, the conus animal, conus species, the particular group of animals, they preferring the small fish because their synthesized or secreted toxins which are helpful for kill their prey, kill and catch themselves. So that way they can easily catch their prey and they can easily swallow it. So that's what that means, just like a hunting. So that's what's very important feature for the gastropods. And the conch, that means the conch gastropods group of animals which feeds on fish and sometimes the annelids worms. And I already told you that they can kill the humans because their poison is somewhat potent. And they have a very special type of radula or radular teeth which are present, and also their stinging apparatus are there. Through their stinging apparatus, the animals, they can target their victim and just transferring the potent toxin into their victims. And also they have muscular food. Similarly, it's most of the animals, they're having the muscular foods. And some of the animals which are present in the land also, it's the terrestrial forms, act as the hermaphrodites. And another remaining all the group of gastropod animals which are present in the marine. And what are the aquatic gastropods which are present? Their acts are involving in the external fertilizations. And although the other biological information such as the nervous system, it's very simple. Small brain and nervous system coordinate their movement as well as their behavior. And bivalves is another, I described their pattern because it's having the two shells. That means two shells which are united together, holding with the very small size of hinge ligaments which are present. That is called as the ambo, which unite the two shells. So it covers the soft bodies, that is called as the periostracum layer, and some muscles open and close because and also in addition to that, it's having the adductor muscles which connect both the valves and unite together. And there are about 10,000 living species, that the living bivalve species which are present. And shells can range in starting from the small size to the larger size. I already told you that 4 to 5 feet of bivalve species which are present in Indian context called as giant clam. And also no distinct head or radula which are present which not present on this particular group, that is in bivalves, because they are the filter feeders animal. What are the microscopic organisms which are present, they can easily filtering through their specialized apparatus or their holes which are present on their animals. And the feeding habitat, most of the sedentary filter feeders, because they are easily settled or attached on their rocky substratum, so they can easily their gills which are acting, which are filtering their microscopic organism for their food purposes, and the waste which are disposed as to their opposite poles. So most of the single pair are gills for the respirations, and gills for the respirations for their trapping minute food particles. And separate sexes, that means they are involved as external fertilizations or in external fertilization point. So most are the marine farms, but many brackish water as well as these freshwater farms, and sometimes in the streams, ponds, and lakes. They are presented very vastly. And coming to the important groups, cephalopods. Cephalopods, as in my dear friends, you know, cephalic in the sense head regions. Their food which are originating from their head regions, that is called the head-footed animals. Most of the complex, most complex of the mollusks, maximum all are marine, and they're having very unique features like tentacles, which are uniformly present on the head regions, and each and every tentacles which having the another specialized apparatus, that is a round like structures which are present, called the sucker, available in the their food, and they are used for just capturing their prey, because the available sucker which helpful for just placing their victim body surface, and they can take it. So that way the suckers which are acting for catching their prey material. And almost cephalopods are purely marine in habits, and there are about 600 living species are there, and they are considered as the fastest marine invertebrates, as well as the brilliant invertebrates groups in animal kingdoms. And this is another important group of important species which are available coming under the cephalopods, that is called as the nautilus. This is almost cephalopods. They are also having the cells, but the shells which are present on the inside of the animals, especially the cephalopod groups of animals. But this is the only species which are available as their shells are externally available, and their other tentacles and other head regions which are comes out from their shells itself. So this is the nautilus species, and also their locomotions. It's somewhat a very interesting feature when we are watching the cephalopod group of animals at the live in conditions. They are the backward movement, just propelling their animals into their backward. The animals, they can easily move. So that's what it's very unique features of the cephalopod group of animals. And almost the reproduction is the internal reproductions. Then their eggs are okay. My dear friends, you can see the things and how the octopus they can opening the box, that means very denser or heavier box, and some of the scientists watching their screen. Yeah, this is very largest octopus species which are available, and you can see their structures at their original habitat. They have exhibit very peculiar color patterns. You can see their different color patterns, which shows the camouflage behavior, and you can see their skins. And my dear friends, and another information, very important information on this particular group. Their skins which consist of number of chromatophores. Even the animal is died. But the animal is died. But up to 24 hours, their cells are live in conditions. They have to exhibit the different colors, and which is very much a source for the production of the carotenoid pigments such as the astaxanthin. So it's very unique features for this particular group. And regarding the conservation point of view, lot of conservation strategies which have been taken by the government of India as well as all over the world. But in my point of view, the conservation strategies, the overfishing and illegal fishing is leaving from our oceans. on the edge, the total collapse. And as the operators look to the plunder, that means stolen or exhausted ocean further. And also the untargeted fishing. This is very common practices by our lovable fishermen. But we should practice, as a scientist, as a researcher, we should practice them for the targeting fishing because some of the other fishes are very economically important fishes, such as the tunas or sardines. They have a special type of nets. They have to catch only the particular group of animal, like the tuna or even the sardines and other animals. So similarly, when we are practicing our fishermen, definitely we should avoid the untargeted fishing practices among the oceans, and we save our resourceful areas from our Indian exclusive economic zones. And also the strengthen the molluscan database. There are a lot of molluscan databases which are available based on their different countries, like in Australia, even in India and other coastal countries. But we should try to strengthen their molluscan database to create awareness among the public and other scientific community to avoid the over exploitations, because the resources are very vast. But when we are regularly catching or when we are regularly utilizing their resources, definitely it leads for the over exploitations, and their biodiversity of this every group should be reduced. And I just show some of the slides from our collection, and you can see the things. This particular for the first picture shows the, and due to their untargeted fishing, lot of journal farms, which including the crustacean, fishes and other sponges, even some lot of the sea grasses and other seaweeds. The untargeting things, they have to wasted our resources. There is no purpose at all, except the producing some other lime or utilizing in the lime industries. There is no other proper utilization of the trashes or the materials. So that's what then reducing. And another picture shows the, and this one, this is shows the Xanthus pyrum or the Turbinella pyrum. You see how much huge amount of Turbinella pyrums which are regularly landed at the Gulf of Mannar regions as well as elsewhere. And throughout our Tamil Nadu coast, they have exploiting these resources. It's somewhat because these are our assets. This is our assets, and most of our assets exported to other countries. And this slide shows the another level of the percentage, and that means another group of organisms, small size of animals, which are exploited by the fishermen, and there is no worth at all. It's only for utilizing the aquaculture industries, just like for making their food or feed. That's all. And my dear friends, I hope my lecture somewhat give the information about the importance of marine mollusks and based on the diversity, how we can utilizing the marine resources or the marine molluscan resources for the production of food or for the production of medicines. Especially in my laboratory, we are dealing with the development of the various drugs from the molluscan group of animals as well as the other marine animals. And with this information, I conclude my lecture according to the best sentence of my Swami Vivekananda. That means our mentor: take risk in your life, because as a marine biologist, we should take risk in your life. And if you are win, you can lead. And if you are lose, then we can guide. So with this information, I conclude my lecture. And on this happiest information, the session or the webinar, this is my duty and responsibility to thank my dear good friend and assistant professor of Department of Zoology, Ambedkar, Dr. Ambedkar Arts and Science College, Dr. N. Tirunavakarasu, for giving these opportunities. And once again, I thank very much for your presence and passion. Thank you.
Thank you, sir. There are, I think you can just take a couple of questions.
Yes.
Can global warming have any genetic mutation or habitat change in mollusca?
Yes, definitely. Why not? Actually, the global warming, that is another problem. Most of the group of the molluscan group of animals which reduced based on, because all the aquatic organisms, they are the very sensible animals. Even starting from a fraction of temperature variations, it leads or it's affect their growth of inverted groups of animals, especially the soft bodied animals like mollusca. Yes, it is possible.
Is there any effect on ocean acidification on the shells of mollusca?
Yeah, I already told you that the very brilliant color. You can see the animal shells. Is somehow they are the very brilliant color. If the ocean acidifications, it may be imply or influence their color patterns, just like bleaching their shells immediately. That kind of characteristics or that kind of what is that unusual phenomena which may be occur on these animals. Yes, definitely it affects those.
Is there any specific medicinal properties for molluscan group of animals?
Yes, my dear friend, actually, this is a pandemic or corona period. Most of our research involving to try to discover the vaccines or the drug for the COVID-19. But the molluscan group of animal, one animals, I already told you that the conus animal called as the conus magus. Conus magus produced peptide that is called as the zincnotide. The first drugs which are derived from the ocean, that is called as the prialt, for potent analgesic drugs which are available in the markets. So the phylum Mollusca shows the extensive species diversity as well as the chemical diversity.
There's one more question, sir. What type of colored material is commonly used for the coloration of pearls?
What
type of colored materials is commonly used for the coloration of pearls?
Yeah, coloration of and the lot of coloring agents which are present for starting from, if you want the bluish in color, you can consider in the phycocyanin, and a lot of coloring agents available. That means approved coloring agents which are available. And for example, if you want to pinkish pearl in this sense, you can utilizing the astaxanthin. Once if you are utilizing the astaxanthin, they can give the very pinkish color to our pearls.
There's one question from the YouTube participants, sir. For avoiding untargeted fishing of mollusk, what kind of gears and crafts we can use?
Yeah, actually, if you are reducing their what is that shoreline, I think shoreline nets are it's very traditional practices or traditional net which are practicing in the coastal areas. If we should avoid the such kind of the nets, definitely we should reduce the exploitation of the marine mollusks, because most of the marine mollusks are the regions are the shallow and shallow to deep, and so definitely we should reduce by using this kind of nets.
The last question, sir. There are a lot of marketing potential for polished shells which are sold in the market. Is it a threat to marine mollusk?
Yes, yes. My dear friend, you see, you faith. This is the God, the Hindu primary God, the first God of Hindu religion, the Lord Ganesha. Whether nature providing this kind of the what is that sculpture? No, man-made material. But you can see how much of the bivalves, gastropod animals which are contribute for making this kind of the shells or the ornamental nature. These are the man-made practices. Nature, they can't provide, they can't, encouraging these kind of the practices. All are man-made activities. Definitely we should reduce these kind of activities. I'm not blaming the Lord Ganesha. I'm blaming only the, the coastal peoples as well as the scientists.
So thank you so much, sir, for clarifying the doubts of our participants. I think we have now come to the end of the session. Over to Dr. Tirunavakar, sir.
Thank you, Dr. Arumugam. Being busy schedule, you have given time to give a lecture on marine mollusk. Thanks for covering the overall spectrum of marine biodiversity and its consideration in Indian perspective. Apart from marine mollusk, how its importance, and you have explained with the live specimens. So this is one of the eye opener to the upcoming, the zoologist and marine biologist students. They could see the animals with different colors and explanations also. And you explained the octopus video and marine mollusk the databases and other things on the conservation strategies. Thanks for giving the lecture to our National Capacity Building Training Program. Thank you, Dr. Thank you once again. Thank you.
Thank you, Dr. Tirunavakarsu and other organizing secretary and co-organizing secretary. Thank you very much.
Thank you. Now, before we move on to the final session of the day, dear participants, here is an announcement. We are coming up with another international conference on the 3rd and 4th of August 2020 in collaboration again with the Indian Science Congress Association of India, International Web Conference on Innovative Technology, Entrepreneurship Avenues, Livelihood Enhancement in Sustainable Management of Animal Farming. The registration link for this is now open. Interested participants can apply for this International Web Conference in association with the Indian Science Congress. Now, we move on to the 4th and the final session of the day. We have here Dr. K. Mahesh Kumar, Assistant Professor of Aquaculture, PG and Research Department of Zoology, Government Arts College for Men, Nandanam, Chennai. He is going to present on the echinoderms in marine region. Now, I request Dr. T. Madhuram, Assistant Professor of Zoology, Dr.Ambedkar Government Arts College, to introduce our next resource person, Dr. K. Mahesh Kumar. Over to Madhuram, ma'am.
Good afternoon to everyone. It is a great honor for me to introduce and welcome our today's speaker, Dr. K. Mahesh Kumar. He worked as an assistant professor in aquaculture, Department of Zoology, Government Arts College for Men, Nandanam, Chennai. He has done his doctoral degree on studies on sea urchin in Kanyakumari, which was the first work in India. He has published several papers in various national and international conferences, seminars, and symposium. He has attended a great number of workshops and seminars and had also been a great resource person in 12 workshops and seminars. He has been specialized in marine biology, echinoculture, aquariculture. He has a hard worker experience as a guest lecturer in Department of Marine Science at Bharatidasan University, as a lab manager in Asian Analytical Laboratory, Chennai, and as an assistant professor in Marine Studies and Coastal Resource Department of Madras Christian College, Chennai. He is very interested in aquarium construction and step up. Now I call upon Dr. K. Mahesh Kumar to share his work with us. We welcome you, sir.
Thank you, Madhuram, ma'am.
Thank you, sir.
I now request Dr. Mahesh Kumar to share his presentation.
Thank you, ma'am, for your. Am I audible?
Yeah, you're audible, sir.
Yeah. So thank you, ma'am, for your kind words. And first of all, I would like to thank the organizers, HOD, Department of Zoology, Dr. Kumaran, and the organizers, secretaries, Dr. Tirunavakarsu and Dr. Saravan sir, and other organizing members. Today, just a minute, I will share my screen.
Mahesh Kumar, sir, your screen is visible, sir.
Yeah. Today, I'm going to discuss about the exploitation and conservation of echinoderms. And so we know from the class 6 you might have start studying about this echinoderms. You know very well about that. So I'm not going to discuss about some general characters, but even though some of the people might have the non-biologists, so for them, we need to give some introduction regarding the characters and the members of the echinoderms family. Right. So what are the general characteristics? The echinoderms are exclusively marine. When you compare to that of the other phylums, if you see the echinoderms exclusively, they are present in the marine environment. And you can see they will habitat in tropical and cold seas also. The term echinodermata, it derived from a Greek word. It means the spiny skin, echinos and dermos. So the echinoderms are characterized by its radially symmetrical, and they are pentamerous. They have no heart, brain, and eyes. But the brittle stars, they have a light sensitive part in their arm in order to avoid the bright lights. And the mouth is situated on the bottom and the anus at the top. And they have a tentacle-like structures called tube feet. So all the echinoderms, they will have tube feet for their locomotion and also for their feeding purposes. Okay, you know about the echinoderms, they will have a well-developed water vascular system which is connected to the tube feet, which act as a locomotor and also for the mechanism to catch the feed. Next, as I discussed about the tube feet and the water vascular system, and you can see all the echinoderms, they will have the umbilical group. In the umbilical group, you can see the tube feet will be situated on the all 5 radially symmetrical arms. And this is the crinoid, the ciliary rays. In each and every crinoid, the rays, you can see the tube feet. And in the sea urchins, you can see all around the test, you can see the tube feet. Right? The respiration is through papillae, gills, and like a respiration tree. Nervous system is absent because it is a brainless organism. Circulatory system is reduced, heart is absent, no blood pigment, digestive system is complete, excretory system is absent. Sexes, monoecious and rarely monoecious, and they are dioecious, agametic fusion and regeneration. So let us talk about the diversity. Globally, there are 7,000 species has been discovered, that is living species, and 13,000 extinct species are now recorded from the fossil record. And from, you can see here, the echinodermata, the record is taken out from the WOMS, World Register of Marine Species. You can see it coincides with the previous slide, 7,444 accepted species of echinoderms all around the world. And in Indian context, you can see Dr. Deepak Samuel, my good friend of ours, he has worked on the checklist of echinoderms, which has published in the Zoo Taxa. You can see around 7,741 species of echinoderms. It comprises of 182 species of asteroids and 138 species of echinoderms and 179 species of sea cucumber, holothurids, and 172 species of ophiurids, crinoids under this echinoderms. Right. So next, the general classification, we have the phylum echinoderm. We have 5 classes like Asteroideae, Ophiuridae, Echinoideae, Holothuridae, and Crinoideae. So likewise, you can see a order of starfish, brittle stars, sea urchins, sea cucumbers, and sea lilies. So this is the general character for Asteroideae. That means starfish. Approximately, we have 1,500 living species, and majority of them found in Indo-Pacific region. They found in majority of the asteroids found in reef areas and seagrass beds. Then found in the bottom, they are benthic, and most of the starfish, they have 5 arms. The mouth is in the oral surface. It's in the ventral part. They have a spiny upper surface, that is aboral surface. Okay. Then they have a mandriporate, which is the opening of the water vascular system, where they get the water for the entire water vascular system, which helps for the locomotion through the tube feet. Right? They have an eye at an end of each arm to detect the light and dark part. This is the only brain which they have, smallest rudimentary nervous system. Like most of the sea stars are brightly colored. They have a very good camouflage in nature. Feeding is with the help of tube feed. They feed on mostly bivalves, sponges, and ascidians. Also, they have scavengers. Also, they have a unique capacity to feed on the bigger the bivalves. They have a capacity to regenerate. And this is the internal anatomy of the starfish. You can see the tube feeds, the gonad, which is found because it's a radial symmetry. All the 5 arms, they will have the gonads. Like you can see the tube feeds extended here. You can have a radial nerve, the tube feed, the papillae. Okay. And this is the exact tube feed, how it sucks. This is a sucking pad. Okay. Now these are all the common the starfishes which you can see, common sea stars. Okay. The Pentaster regularis, Proteaster lymphi, and the Lynchia multiflora and Lynchia vivigata. And next go to the Ophiuridae, that is brittle stars. They have a central disc with 5 whip-like arms. The Ophiurides means snake. They will move like a snake. No anus, only waste removal to mouth. They are highly agile and hides in the crevices during the daytime. Only they will emerge in the night time to feed on the planktons, some of the benthic planktons. And some found intertwined in the coral branches, feeding the mucus of the corals. No eyes, only they have a photosensing the plates which is present on the top in order to avoid the excess rays. Sexes separate, fertilization external, larval cells nucleus, asexual reproduction by fragmentation, locomotion using arms, not the tube feed. Here they won't use the tube feed, only the creaky movement. Okay, they will move very fast. Then they are scavengers. They have an escape mechanism through fragmentation. Whenever is there any threat, they will simply remove their arms. They will go like the lizards. Then high degree of regeneration. This is the anatomy of Ophiurides. So you can see the snake-like the arms and the oral plates, and you can see the entire tube feed runs over all the radial canopies. And these are the common Ophiurides. You can see it in India, the Amphipolis stomata and Ophiotella leni. And Echinoideae, over 940 species of echinoids distributed worldwide, and their habitat in the intertidal up to 5,000 meters. There are 2 echinoideae groups. One is regular and other one is irregular. Regular is our sea urchins and irregular is our star heart urchins. Okay, so the regular urchins, they are found in rock and substrate. Irregular echinoids are the sand dollars, are generally found in the soft grounds like the sandy beaches. They are pentaradially symmetrical water vascular system. They have well developed water vascular system, and the sea urchins, they will have a combined fused ossicles which is formed as a test. You can see in the beaches the washed out test you can form. This is not an entire sea urchin but only the test part. That is a skeletal part, right? They have a peristomial, in the aboral region, they have a peristomial membrane. In the peristomial membrane, they will have 5 the jaws, that is called as horizontal landing. It's a powerful chewing apparatus. Their mouth leads to the intestine, and the anus is located at the center in the aboral surface of the regular echinoids. The spines and the tubes used for the locomotion. Then on the aboral side, the tube feed function is respiration and sensation. So in the sea urchins, the tube feed is present around the peristomial membrane, which acts as the respiratory organ and also for the sensation. So this is the internal anatomy of the sea urchin. You can see here the test, the spines, and in between the interambulatory region, you have a small spines in the small holes. Through the holes only the tube feed emerges outside. See here, it runs all along the test. Okay, so they have a very simple digestive system, leads from the mouth and to the anus. Okay, around this is a coil intestine, and in each and every the ambulacral plate they will have 5. So each ambulacral plate they will have the gonads. Okay, 5 gonads they will have. So this is the horizontal lantern, very powerful jaws, which is operated by 2 muscles. Okay, and this is how they will habitate in the rocky region. And this is the common the sea urchins which we can find it in our Indian coast, like Stomophosus variolaris, Salmacis bicolor, Salmacis virgulata, Temnoplurius tauromaticus, Trypnotus, Toxoplastis pylorus. This echinoidea, sea urchin, which is have toxin in their spines. Then Trypnotus gratula. See, this Toxoplastis, they have a capacity to shoot the spines. Whenever if you are going to the collections, they will shoot the spines with the toxin. So you get a little bit irritation, and it will goes off in a smaller little time. Then this is the irregular urchins like Clypeaster, Echinodiscus, and this is the heart urchin, sand dollar, and heart urchin, Lovinia, elongata, which is commonly available sea urchins, sand dollars, and heart urchins of our Indian coast. Then next, the Holothuridae. See, the Holothuridae or sea cucumbers have 110 described extant species, that means living species. They are found all over the world, and with the shallow waters to the deep flows, we can found they are benthic. Okay, few are pelagic, and the color varies from brown, olive green, and they varies from 3 centimeter to 1 meter long. They have a worm-like structure. Okay, they will look like a worms. Okay, but they are also retaining the penta-radially symmetrical characters of echinodermata, spherical in body shape, they are wrong. Okay, mouth and anus are located at the opposite under both the poles. Right, 5 rows of tube feet also available, and along the cylindrical body, they have 30 branches of tentacles around the mouth. This is what we can see like our radish, or it looks like a cucumber, so it is called as a sea cucumber. Right? The tentacles are actually a part of water vascular system. The important thing is this respiratory, which comes out near the rectum. It is on the opposite side of the mouth where it is used for the respiration. So in some of the animals, you can see the escape mechanism of sea cucumber is they will eject some parts. So mostly they will eject this respiratory trees, okay, which will be called as the Cuvierian tubules, okay, which they eject outside so that the predator they will eat this Cuvierian tubule. They will, okay, and this is the anatomy of sea cucumber. You can see this area only will have this Cuvierian tubules, okay, and this is the respiratory tree. So this is the respiratory tree. They will have tentacles and very simple digestive system. Like in gonad, you can see they will have the 5 gonads. This is a common Holothuridae, which you can find it in our Indian waters like Scabra, Holothuria spinifera, Holothuria atra, and Stichophus hermani, okay, which is the common sea cucumbers which you can find it in our Indian waters. And next is the Crinoidea. Very the smallest class, only 600 species available in this Crinoidea. Mostly they are deep sea, common on coral reefs, which is called as sea lilies. Okay, commonly. They have 2 specific body parts, like they have a calyx and the ray. The calyx is like a cup shape, and it is a central portion which bears the rays. Okay, they have anus and the mouth on the oral side. They have rays along or long arm-like extensions in the calyx, which will be used for feeding. Some of the deep crinoids, they have a 3rd body part which is called as a stalk, which serves to anchor the crinoids to the substratum. And another conspicuous feature is they have the cirri. The cirri is the only part which go and cling the substratum. And the unstalked crinoids, they have cirri on the backside of the calyx. Okay. The cirri stalked crinoids are extended from the stalk. The function is adhesion. That means they cling to the surface. The crinoids are the filter feeders. They use the tube feet to capture the food, and they will make a string-like string, and then it passes through the groove and it moves to the mouth. Okay. The rays of the crinoids are important for locomotion. They will move like a bird, like this. Okay. The ability of swimming is very slow through the water because they will using this rays to move all along the water. So this is a common crinoid which you can find it in our Indian water, the Trophimetra cardia. So this is the anatomy and the exact picture of the crinoid. You can see this is the calyx, the arm or the ray, and you'll have the pinnules. Then this is the cirri and this is the stalk. Okay, and you can see the central radial disc will have the mouth, interambulacral area, the ambulacral grooves where you can see the tube feeds. Here you can see the tube feeds which run over the ambulacral grooves. Okay, and you can see the food particle which move through the groove to the mouth. Okay, and you can see the rays here. This is a start crinoid. And next, the important part that we have to discuss today is the role of echinoderms in ecosystem. So everybody, they know what is echinoderm, where they are living, but we should know what is the exact role in ecosystem. See the asteroids, the starfishes. So what is the role? Mostly they are the top predators and they will act as a keystone species. So what is a keystone species? The keystone species is an important animal or a part of an ecosystem. Without that species, the ecosystem won't exist. So if you take starfish, you can mostly find it in the rocky shore areas. They are controlling the the organisms such as mussels, barnacles, and then the skyton. If the starfish is removed, you can see the abundant growth of this mussel will occupy all over the area, and they won't allow the other animals to grow in there. So you need this particular starfish to control all this interesting organism which grows over or occupy with a little time they will occupy more in that particular ecosystem. So the starfish here is act as a keystone species to control these mussels and the chitons and the barnacles, etc. So when you control these things, then automatically the algae will grow, the sponges will grow, and also the ecosystem will flourish in that particular area. They'll form a niche, very well made niche. So here you can see this is a small experiment which I got it from the net. So the keystone predators, the pain disaster is exclusion experiment. You can see the mityless mussel outcompeted the other space holding species, reducing the species diversity from 15 to 8. The starfish prefers to feed on the dominant mussels and barnacles. So this is how they are dominating the ecosystem. And also, you can see the Acanthaster plantae, you can see the crown of thorn, have increased in the population and damaged the coral reefs as a result of their grazing. So many asteroids are the top predators and keystone species, but the Acanthaster plantae is very dangerous to the coral reef environment. Okay, so we have to check this Acanthaster plantae. There are also a very good part of this starfish also available, and other bad thing is this, only one is this Acanthaster plantae. So next, Ophiuridae, if you see the role, they are reshaping the sediment in the ocean floor. Because of their movement, twist and tweaking movement, they will be called as the ecosystem engineers, seafloor ecosystem engineers. And the brittle star, normally you can see their twist and coil, which affects the distribution of the other species in the coral reef and also in the sea fans. And one of the interesting thing when I come across is, they found this brittle star helps to brush the smoothing or flop of the branches of sea star simply by extending their arms to the feet of drifting bits of food, which see this particular picture is taken up from an area where more of this oils has been expelled. You can see the entire oil is smoothed this particular coral or the sea fan. You can see the brittle stars cling over, they will prevent this clogging. And also, when they move all along this the sea fan, it removes all the clogging and makes the sea fans to grow faster. So in this way, the crinoid is the Ophiuride, the brittle stars, are preventing this particular the corals and the sea fans from smoothing of this oil. And next, here comes the important the species like the echinoid, the sea urchin. They act as a keystone species in the seagrass environment. You can see, I will explain in a simple way. When the sea otter is the species, because the sea otter is a major predator which feeds on the sea urchin. When this the population is decreased, the sea urchin population will increase, and they will graze upon the seaweeds, and you can see the parent lands. And one point of view, here the parent lands will help more on settlement of the coral reefs and other sedentary animals to grow over here. But in other point of view, you won't get any the seagrass community grow anymore. If at all only one population, you can see the sea otter get reduced, and automatically the sea urchin population will increase, and it decreases the population of the seagrasses, and it increases the barren land for the growth of other sedentary organisms. And now, in turn, if you have a more fishing pressure on the sea urchin, and you can see the available space for the other sedentary organisms will get reduced because of the overgrowth of this sea weeds. Then, if both the things are decreased, and you can see the decrease in the fisheries, and the top predator species like the killer whales also get reduced because they mostly feed on this sea otters and otter, the Pinipedia, the sea cows, sorry, walruses, seals, etc. Now, so in turn, if you see the increase of sea urchin, which decreases the seaweed environment, seaweed population, and also increases the barren land, which in turn reduces the fishing, total production, and also it reduces the population of the higher predators. Now next we will go to the Holothuridae. See, the Holothuridians, the sea cucumbers, they largely feed on detritus feeder, and they clean up 90% of the benthic environments. They are the major portion which contributes in the bioturbation in the benthic region. It refers to rework or stirring or mixing the sediment layers by these organisms. And bioturbation of the sediments in the reef, lagoon, and seagrass meadows has a numerous physicochemical efforts and sediment permeability, water content, which in turn increases the oxygen concentration to improve the sediment permeability. See, many commercially exploited sea cucumbers, they inhibit in this soft habitat such as coral reef, lagoons, seagrass meadows, and mangrove systems, and deep continental shelf habitats. See, why I'm insisting this is when you're in the next coming slides, you can see how it has been get affected. The sediment cleaning and nutrient cycle is the major part of the Holothuridae or sea cucumbers in the ecosystem. And now you can see how the bioturbation and the sediment cleaning is happening because of this sea cucumber. It literally works like an earthworm. And now you can see the nutrient cycle, how it cycles the nutrient. See the macro bacterial detritus, they will uptake and they will excrete as the phosphate and the ammonia. Then the crinoidea, which forms a very little part in the ecosystem, but it only forms as a diet for many crustaceans and the fishes. Now we will go on to the important part, like how these echinoderms are exploited. You can see the total production of the sea cucumbers in 2018. You can see it in the tons. It's about 48,000 tons, and Chilean sea urchin is 32 tons, and the Strongylo centaurus species, you can see it is around 25 tons in the 2018, which has been recorded by FAO. So how much echinoderms we have exploited? See, and we are exploiting these echinoderms for so many purposes. One of the important purposes for the aquarium industry. You can see many marine invertebrates, including echinoderms, are popular in the aquarium trades. In Mexico, Indonesia, Singapore, Fiji, and Sri Lanka are the main exporters. You can see this, how the starfish are kept in the aquariums along with the other fishes. And next for is both the sea stars and the urchins as a souvenirs. If you go to any beach, the places, you can having this shell socks. Okay, example Rameswaram, Kanyakumari. Okay, wherever you go, you can see the these starfishes in the shells of as a souvenirs. You can see how many. Lastly, our my good friend Armanan has insisted, like the conservation part. You can see how many seashells along with the echinoderms also they are saved. Okay, see. So this is only a part of a photograph, but if you see, whenever you go and take any shells off, they will have a large amount of this starfishes. See here, even the sea urchin, my PhD, I made PhD in this species. You can see the spines here. So they will use the spines to write in the slate. It is in Tamil they will call it as a kuchi. Okay, so they will use it as a pencil in writing in the slates. You can see how many these starfishes are literally lying here as a souvenir. And here you can see the sea urchins. This is a Stonefnoses variolaris. It's normally used to, this sticks, the spines they are used to write it in the slate. You can see the starfishes here, even corals, all other chants, the spider branch, and the elephant branch. Even you can see the corals. And these are all some of the other the utilization of the sea urchins like lampshade, the for lampshade and also for the ashtrays and the mirror, etc. And another interesting thing what which I refer to the Vivek, he told only the small lemon it will save our the big mechanism or big lorry. You can see this is used for drishti in Tamil Nadu and south part of India. We are using along with that we are also using the starfishes and the shells. You can see. I don't know how it rectifies our bad thing. I don't know, but even though we are using this echinoderms in the for relieving reducing the evils entering into your house or vehicles, etc. Then the sea urchins, they are used as a food. You can see the it's a delicacy. The roe, no, the gonad. Okay, it is delicacy and it is used in Japan in the sushi bars. And sea urchin uni is primarily sold as a fresh rather than frozen. They have very sweet and flavor. It melts in your mouth like your Cadbury's. And sea urchin raw available in the market. You can see this individual rows. They are packed nicely packed, like how the Swiggy and other people are delivering the fresh meat. Likewise, they are delivering. Okay, you can see it is cost around a rack cost around $19. This is the sushi and the live sea urchin also. You can buy it for taking this. So this is how it looks like the golden color. So this golden color it fetches more price in the market. So approximately the large consumer of this sea urchin roe is Japan. Okay, the roe means it is nothing but a gonad, both male and female. You can see how nicely they are packed. And the fresh roe from China, North Korea, and USA cost respectively. You can see here the cost. And Tokyo is the largest market. And the A-grade roe cost around $15 for 18 grams or 80 grams. And I did some research on that, like the culture of echinoderms. You can see. So it can be done by induced spawning. So the egg and the sperm will get released, the larval development there, larval setting, and also you can see to feed the larval young ones, live feed culture and the mass culture has been made. And you can see the development stage of the sea urchin. Like this is a fertilization stage. You can see the sperms around here, fertilization membrane, and the cleavage, 2-cell, 4-cell, 8-cell, 16, 32, and morula stage. And also now you can see the hatching of morula, free swimming stage, free swimming blastula, rhizome stage, luteus, 4-arm, 8-arm, and the settlement metamorphosis stage. And this is the culture setup which I made to culture the sea urchin by using 3 diets. Okay, so this is how I made an attempt to made this urchin as a commercial one. So if you know the hatchery technique, then automatically you can utilize this particular situation for the export purposes. Now let us come to exploitation of this holothurides. So here in India and all over the world, the holothurides are very much expensive. So why? You can see every day and now and then you can see in the newspapers, you can see such type of news like tons of sea cucumbers were seized. Why? And you can see the sea cucumbers are seized in Nagapattinam, and also the poaching of sea cucumbers, and it goes to Sri Lanka and it through Lakshadweep Island, and high demand in Eastern Asia, the poaching of endangered sea cucumbers, and this is particularly Express News from 2 So why? And you can see an illegal trade that thrives with the sea cucumber. Mainly you can see they are contributing much from Gulf of Mannar region. Why? Sea cucumbers are valuable than that people are risking their life diving for them. You can see a kilo can cost around $3,000. That's why most of the people, they are more concentrated on the sea cucumber rather than this sea urchin. Now, for this, the China, they have set up the world's largest sea cucumber farm. And also you can see they have also studied about the nutrition part. Seagrass shows the potential in the growth potential in sea cucumber varieties. And then why this holothuridae or sea cucumbers are more important? If you see, it is used in the traditional medicine, the Chinese traditional medicine. When I have gone through this, you can see it rectifies more it is used in the treatments like weakness, impotence, disability, disability of age and constipation, intestinal dryness, frequent urination. Then they have the other properties also. You can see they improve the frequent urination and kidney deficiency. Sea cucumber is cooked with mutton for nourishing essence and blood in persons who suffered from emaciation combined with the soup with the float. You can see so much treatment is available in the traditional medicine in China. So that this sea cucumber has been poached and through Lakshadweep it goes to the Chinese market. You can see mostly it is used for weakness, impotency, and for age, in order to reduce the age part. And there is also the research evidence also available. You can see most of the sea cucumbers, they have the mucopolysaccharides and chondroitin sulfate, which used for arthritis and osteoarthritis. Okay, and sulfate and mucopolysaccharides and chondroitin for inhibiting the virus. And Japanese have patented the sea cucumber chondroitin for HIV therapy and also for herpes viruses. And even the Russian, Japanese, Chinese, they revealed that sea cucumbers also contain the saponin, triterpene glycosides. They have used, it is similar to ginseng. It's an energy compound. It's a root of a plant, ginseng, and it is having the anti-inflammatory and anti-cancer property. So such a valuable thing, this sea cucumber. That's why they are poaching it. And these are all some of the other utilization, like in biotechnology. You can see the compounds like triterpene glycoside. It has been recorded in 19 holothuridae, which is having the cytotoxicity activity, and glycosides as growth inhibition of bacteria and fungi. Leptin, they have cytotoxicity in the human lungs and the cancer cells. Then fucosylated, chondroitin sulfated, they will promote the proliferation of blood in the vessels to prevent the arterial thrombosis. And also this fucosylated chondroitin sulfate in sea cucumber, which has been used as alternative for heparin. They are also possess the anticoagulant activity. And these are all the references, so that only it is having such a demand in the market. And also you can see their uses in HIV and arthritis. And also echinoderms may become a powerful, useful in bionics, because now the recent research has been made that thousands, they have the brittle stars, they have the lens, the focus light on the nerve bundles that run behind them, that presumably receive signals to the further process. So these the crinoids, they will have a single cell calcite crystals which will act as a lenses. So now thousands of lenses from a compound eye that covers the upper surface of the animal, resulting in a function similar to digital camera. So now they are figuring out how this animal is getting away from the light. So that research is going on in that. Then the echinoderm embryos and larvae has been used as an experimental model. So it's an age-old practice, like these embryos has been used as a model for the development of biology and also other genetic evidence. And now we are concentrating on the conservation. And as a Indian part of view, we have kept all this sea cucumber, only the sea cucumber in the Schedule 1, which comes under the Wildlife Protection Act. So all the echinoderms have been illegal. Okay, so no sea cucumber has to be taken up from the Gulf of Mannar or any biosphere reserves or any Indian waters. Then it will land to they will have, it's an illegal process. Okay, along with this mollusca and also with the coral. And if you take the other the conservation measures, like Lakshadweep gets the world's first sea cucumber conservation reserve to curb the smuggling, because through Lakshadweep only they are smuggling the sea cucumbers to the other part of the world. So they are now the very strict measure has been taken up in the Lakshadweep area. And if we take all over the world, the sea cucumber production is more and fishery is more. What they have taken up, like the closed season during the time of spawning, like our fishing ban, they are also doing the same thing, like the closing of season and gear restrictions, destination of no take, okay, then marine protected areas, daily catch limits, and minimum legal size, and prohibition of night fishing, and restriction of use of scuba for harvesting. So these are all the measures which has been taken internationally in order to conserve these echinoderms. And also the another alternative method, what in near future what we have to do is to develop a laboratory culture method. That's why in my research also I have taken up this laboratory methods in order to laboratory techniques and to study the hatchery technology and grow up. I made on sea urchins to enhance the commercial importance. And sea urchin culture has been practiced in Japan for many decades. Okay, and whereas in the case of the sea cucumber, it is very old, like 1930s they have started. In 1950s they have started producing the first production. And mass cultivation of Holocaust scabra was also established in India, Australia, Indonesia, Maldives, and Solomon Islands. So this is another alternative method how we can conserve. That means the production through the aquaculture, okay, culture methods. And this is the another important the news what the sea urchin hatchery has been set up in the coral reef environments in order to prevent the growth of the excessive algae or invasive algae. And this is the same thing, like the scientists now breed urchins to test the export potential, okay, in order to get the international attention towards this particular species of a high demand one. And the another thing is the CPEC has developed the entire life cycle of Holocaust scabra. So they are successful in production of, these are the handout of the CPEC about their hatchery operations and successful in their technology. You can see what is the sandfish. The beach steamer is the famous product which has been produced by these sea cucumbers. Okay, dried sea cucumbers, which will be consumed as a soup. Okay, and this is how they are standardized their technology. Now they are transforming the technology to the other states. So with this, I conclude my seminar, in the conservation and exploitation. Thank you.
Thank you, sir.
Thank you, sir.
Questions? Can these echinoderms act as ecological indicators for pollution?
Yes, sir. Some of the echinoderms, they will act as indicators like these starfishes. So they acclimatize themselves to a particular pollutants, and they can thrive along that environment. So it can be used as a pollution indicator also.
Any medicinal properties for echinoderms?
Any
medicinal properties?
Yeah, that's why I don't know this sea urchin roe and holothuridae, they have a number of the medicinal properties. Even the sea urchin roe, they have a capacity to, it has been used as, in some of the research, they have told that as used as an aphrodisiac, and it has been reduction, increasing the potency, and also reduces the piles. Likewise, they have, but there is no base data for that. But holothuridae, they have all data regarding this.
What is the relevance of sea cucumber exploitation?
What is the
relevance? What is the relevance of sea cucumber exploitation, sir? We're asking why it should be exploited, the need for its exploitation.
Yeah, need for exploitation is in, see, it is having a wide range of traditional medicinal properties, but not, it has been used in India, but in Chinese medicine, they have a wide application in there. As I mentioned in the slides, they have a number of, it cures all the ailments like osteoporosis, and they are treating for kidney, mainly for arthritis and kidney they are treating, and also to reduce the aging properties. So these people, because it is cost more in the market, so they are exploiting it. Illegally, they are exploiting in India, but whereas in other countries, legally they are exploiting.
What is the effect of toxins secreted by starfishes? Do they affect the organisms inhabiting nearby them?
The starfish, I don't think so. They will have any toxic effect. Only the predatory effect only be there. Okay. So I didn't come across any the toxicity over the fishes like the sea urchins and other toxoplasmosis. I don't think so. They have any toxicity, but they have a predatory in nature, highly carnivorous. Like this Acanthastra plantae, they will even entirely they will clear out all the coral reefs. If hundreds of that, they immediately will clear all the coral reefs.
Thank you, Dr. Mahesh Kumar, sir, for your excellent presentation. Over to Dr. Tirunavakkar, sir.
First of all, I would like to thank Dr. Mahesh Kumar having accepting our invitation towards our National Capacity Building Training and Biodiversity and Conservation. Dr. Mahesh Kumar has covered sea urchin, sea star, and other echinoderms with colorful pictures, and how it's used as a ketone species and edible food purposes and all. And he also covered the exploitation and the conservation measures to be taken. And for the upcoming researchers, I want to convey here. This is the echinoderms, which is not it's untouched area, especially for the taxonomy and the culture aspects. Dr. Mahesh Kumar, I think in 2006 to 8, he stayed at Kanyakumari. He took the samples and he practiced the echiniculture, the sea urchin culture practices. First time in India, he published the paper also. So this is one of the scope area in which the budding researchers, you can do many works for the upcoming years. Thanks, Dr. Mahesh Kumar, sharing your views and your experiences in this platform. Thank you.
Thank you, Dr. So in this answer, I would like to thank the head of the department, Dr. Kumaran sir, and also the organizing secretary, Tirunal sir and Saravanan sir, and all other staff members of the Department of Zoology, Government Arts College, Dr. Ambedkar. Thank you. Thank you very much, sir.
Dear participants, we have now come to the end of the first day session. Thank you for staying with us. Here is a small announcement for tomorrow's session. Tomorrow morning, we'll be meeting again by 9:50. So tomorrow's theme is diversity and conservation of marine vertebrates. We have marine ornamental fishes, sea turtles, and marine mammals presentations. The presentations will be by Dr. T.T. Ajit Kumar from Cochin, Dr. Kakoli Banerjee from Odisha, and Dr. K. Madhu Mahesh from Dehradun. So please stay connected with us tomorrow. So we'll meet again tomorrow morning by 9:50. As far as the sessions for today is concerned, we will not have any feedback for the day. We have just noted your attendance. All the participants in the Zoom and all the participants in YouTube, we have noted down your attendance. So attendance is mandatory for all the 3 days. We have just noted down all your presence here, and the feedback will be shared later. So tomorrow morning, stay tuned with us at 9:50. So we'll meet again at 9:50 tomorrow morning. Thank you.